warm welcome to you all to the 7th Annual National Conference of Midnapore City College, MCCCON 2026, centered on the theme Trends in Synthetic Biology and Sustainable Development, being held on 4th, 5th, and 6th February at Midnapore City College. The conference provides a distinguished platform for scholars, researchers, scientists, and academicians from diverse disciplines to come together and engage in meaningful dialogue on the transformative potential of interdisciplinary collaborations and promote ethical scientific thinking. Once again, I extend a heartfelt welcome to all our honored guests and participants.
Good morning, everyone. I extend my hearty welcome to everyone present here. With this, we shall now proceed to the inaugural ceremony. Now, may I respectfully invite Dr. Pradeep Ghosh, Honorable Director, Sir, Midnapore City College, to kindly grace the stage. Sir, please. I now invite Dr. Soumya Shankar Sarangi, Chief Medical Officer of Health, Paschim Medinipur, to kindly come to the stage. May I now request our Honorable Director, Sir, to felicitate him. A warm round of applause, please, everyone.
A big round of applause, everybody. I now respectfully invite Dr. Sudipto Chakraborty, Principal Medinapore.
Big round of applause, please, everyone join us. Big round of applause, please, everyone join us. Thank you, sir. Now I request our scholars to felicitate our other esteemed dignitaries and guests of the stage. Now I would like to request our scholars to felicitate Professor Nirmal Kumar Sarkar, former Professor, Presidency University, and Senior Professor, Department of Biological Science, Midnapore City College. A big round of applause, please, everyone join us. Thank you, sir. Now our research scholars will be felicitating Professor Dilip.
A huge round of applause, everybody.
Thank you, sir. To begin the day on a note of melodious grace, we now invite our students to present the inaugural song.
Hello, hello, hello. Hello, hello. Hello, hello, hello. Hello, hello, hello. Hello, 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 hello. Hello. Hello, hello. Hello, hello. Hello. কিলাবন্যে পূর্ণ প্রাণ প্রাণী সোহে আনন্দ বসন্ত সমাগমে কিলাবন্যে পূর্ণ প্রাণ প্রাণী সোহে বিকশিত প্রীতি কুসুম হে আনন্দ বসন্ত সমাগমে বিকশিত প্রীতি কুসুম হে বিকশিত প্রীতি কুসুম পুলকিত চিত কাননে কিলাবন্যে পূর্ণ প্রাণ প্রাণী সোহে জীবনলতা অবনতা তব চরণে জীবনলতা অবনতা তব চরণে হর্ষ গীত উচ্ছ্বসিত হে আনন্দ বসন্ত সমাগমে হর্ষ গীত উচ্ছ্বসিত হে হর্ষ গীত উচ্ছ্বসিত কিরণ মগন গগনে কিলাবন্যে পূর্ণ প্রাণ প্রাণী সোহে আনন্দ বসন্ত সমাগমে কিলাবন্যে পূর্ণ প্রাণ প্রাণী সোহে নমস্কার
Thank you to our students Neha, Divya, Orimita, Mrigakshi and Anesha for their beautiful and soulful performance. We now invite our respected dignitaries to initiate the watering of the plant ceremony, symbolizing our collective commitment to sustainability and a greener future. Sir, please. A big round of applause, please. Everyone join us. May I now request our dignitaries to kindly come on the stage for the abstract book opening ceremony. A big round of applause, please.
A big round of applause, everybody, for this auspicious moment.
Now we would like to request all our honorable dignitaries to take their seats off stage. Now I would like to request Dr. Soumoshankar Sarangi, CMOH, Paschim Medinipur, to share his valuable speech with us. Over to you, sir, please.
Good morning. A very good morning to the every student present here, every dignitary is present here. My dear principal, Sudipto Chakraborty, Vice Principal Kuntal, Founder Director Pradip Ghosh, other Professor Dilip Nandi, Professor Siddhartha Chatterjee, Professor Nirmal Ghosh, and Professor Bhavatos Das, sorry, and all my faculties here, teaching, non-teaching staffs and students present here. It's really a memorable day for you. I'm very much happy to be part of it, that the 7th Foundation Day, MCCCON 26, is going to be held here from 4th to 6th with a theme, synthetic biology and sustainable development, for which this abstract book has been opened by us. I'm very much proud of this institute, Government of India, in different research projects. I was told of it. They are promoting the research, and young minds here, they are nurturing with a vision for doing something for the innovative for the society. And it's a really memorable day that I present here in this occasion. And I think that the students passing here, they used to get support from our department whenever we asked. We have got collaboration with this college in different course curriculums, like biomedical lab science and also other and different category of students used to come to our health facilities for their practical experience. And we are happy to extend support to the needy students of this Midnapore. we have started with a very good song, Eki Labonne Punno Prano. Very, really they sang well, and the anchoring is awesome. I have attended several college programs, but it's really nice to have such quality anchoring and quality inaugural song in your college. And it's the vision of this college, I think. I was told that they not only nurture the education, they also nurture the cultural abilities, the sports, the social activities, community outreach program, whatever maybe. And they use this platform to build a holistic student, not only in education, but also a social responsibility and having other talents. It may be sports, it may be culture, it may be writing, everything being nurtured here. I just have seen the abstract book. It was so nice. Really nice to have in this college such amount of research are going on, and they are publishing in the national international journals, which you can't find in any other college, I think, in this Midnapore town. It's really very proud of this. The faculty is here, director himself, they are promoting research, publication, quality training, and they have built a really good infrastructure. I really feel proud of this kind of conference hall, this kind of laboratory you are using. The infrastructure of the laboratory is just kind of international standard. And I extend my heartfelt gratitude to the college authority for giving us opportunity to holding the examinations, recruitment examinations here. And we also use this infrastructure for several occasions. And lastly, I hope that after passing, you will uphold the legacy, uphold the excellence of this institute in future.
College to kindly come to the stage for the welcome and the keynote address.
Honorable Founder Director of the college, Dr. Pradeep Ghosh, respected CMOH of Paschim Medinipur district, Dr. Soumya Shankar Sarangi, respected Principal, Dr. Sudipto Chakraborty, respected Vice Principal, Dr. Antal Ghosh, respected Professor Bhabatosh Das of Greek Translational Health Science and Technology Institute, Faridabad, Professor Sandip Chattopadhyay, Head, Biomedical Laboratory Science and Management, Vidyasagar University, other distinguished resource persons and guests from different institutes, learned colleagues and organizing committee members, dear staff members, researchers, and student participants. Very good morning to everybody. It is my proud privilege to make a prelude in this inaugural session of the 7th Annual National Conference of Midnapore. At the outset, I express sincere gratitude and extend Hearty welcome to you all for your active participation in this conference. I also express our indebtedness to the CMOH of Paschim Medinipur district, Dr. Soumya Shankar Sarangi, for his kind deliberation in this inaugural session. Now I would like to brief you all, especially the students, about our college. The foundation stone of this college was laid on 6th February 2017, and the college started functioning in a single two-storied building on 10th August 2017 with just 134 postgraduate and 20 undergraduate students. But massive changes took place in the scenario in the last 9 years. These changes are primarily due to the untiring efforts, enviable tenacity, and farsightedness of Dr. Pradeep Ghosh, who is not only the director but also the real cardiac pacemaker of this college. As compared to a single two-storied building in 2017, the college now spans across 3 multi-storied academic buildings accompanied by a few smaller annexes. Besides, a central library started functioning since the last year, and the construction of a multi-storied humanities building is in progress. Again, as compared to only 154 students in 2017, the student strength reached a total of 7,009 in 2025, which included 4,367 undergraduate and 2,642 postgraduate students. The college now teaches 19 postgraduate and 17 undergraduate courses under Vidyasagar University, along with 4 postgraduate and 2 undergraduate courses under West Bengal University of Health Sciences. Moreover, this is the only college in the 2 Midnapore districts which teaches different job-oriented UG or PG courses on agriculture, dairy, MFSC aquaculture, hospital administration, medical laboratory technology, radiology, medical microbiology, nutrition and dietetics, computer application, etc. It's my pleasure to mention here that 6,758 or 96% of the total 7,009 students are receiving one or another kind of scholarship from either Government of West Bengal or Government of India. Since 2021, above 100 of our postgraduate students qualified NET, SET, GATE, JAM and JEST, etc. research entry tests. Moreover, some of the alumni of this college ensured their placement as researchers in reputed institutes of South Korea and Germany, in addition to IIT Mumbai, IIT Delhi, IISc Bangalore, and IISER Kolkata. At present, we have 25 well-equipped laboratories containing various research instruments. Since December 2021, Vidyasagar University has sanctioned PhD Research Center status to our college. Till now, 50 research scholars got enrolled in Vidyasagar University for their PhD pursuit under the supervision of different teachers of this college. 34 of them get scholarship from different funding agencies, while the rest get monthly Moraine Research Fellowship from the college itself. Till now, 7 scholars have already submitted their PhD thesis from this college to Vidyasagar University, and 2 of them have already been awarded their PhD degree. Again, Dr. Sabyasachi Pal of Physics Department and his scholars recently discovered 34 giant galaxies in the space from this college only. Their groundbreaking discovery received wide coverage by the weekly newsletter of the NASA, as well as different newspapers and news channels of the whole country. Moreover, different teachers of our college, especially Dr. Sudipto Chakraborty, Dr. Kuntal Ghosh, Dr. Shravani Pradhan, Dr. Suchishmita Roy, Dr. Sabyasachi Pal and Dr. Shantanu Kaur Mahapatra have been able to publish their research papers in different high impact factor journals in the last few years. We gladly add here that 6 talented teachers of our college are utilizing grants over 5.02 crores of rupees from different funding agencies for conducting their research projects. To mention them chronologically, they are Dr. Shravani Pradhan, the coordinator of nutrition science, and also the organizing secretary of this conference. Dr. Kuntal Ghosh, former postdoctoral fellow in Jeonbuk National University of South Korea, and now the vice principal of this college. Dr. Suchishmita Roy, a diligent and sincere teacher of nutrition science. Dr. Shantanu Kaur Mahapatra, head Department of Paramedical and Allied Health Sciences. Dr. Sudipto Chakraborty, former postdoctoral fellow in Rush University Medical Center and Masonic Cancer Center of USA, and now the principal of this college. And number 6, Dr. Shubhendu Kumar Manna, an assistant professor of physics in this college. Now turning all your attention to this 7th annual national level conference of our college, sponsored by ANRF, Government of India. The theme of this year's conference is Trends in Synthetic Biology and Sustainable Development. Synthetic biology is an interdisciplinary field that applies engineering principles and computer technology to biology to design, construct, or modify biological parts, systems, or organisms for useful purposes. On the other hand, sustainable development means meeting present needs without compromising with future generations' ability to meet theirs, along with balancing economic growth, social well-being, and environmental protection. Synthetic biology may play a key role in ensuring sustainable development because synthetic biology involves redesigning organisms to have new abilities, such as producing safe vaccines and therapies, improving nutritional value of crops, mitigating environmental pollution, creating eco-friendly pesticides, fertilizers, and biofuels. In the present conference, valuable deliberations will be made by a galaxy of invited scientists on the exploitation, benefits, and prospect of different synthetic biological approaches. In addition, 20 research papers will be presented orally by research scholars and teachers, while 40 posters will be presented by the enthusiastic students of different institutes. All the eminent scientists to make valuable deliberations in this conference are, simply speaking, stalwarts in their respective fields of research. I just mention them in an alphabetic order. First comes Professor Bhadutala Das, an eminent professor cum molecular biologist, a microbiologist from BRIC Translational Health Science and Technology Institute, Faridabad. Then Dr. Bimal Prasanna Mohanty, former Assistant Director, Inland Fisheries, Fisheries Science Division, ICAR, New Delhi. Then Professor Dev Prasad Chattopadhyay, former Founder Director, ICMR, National Institute of Traditional Medicine, Belagavi, Karnataka. Professor Sagarmoy Ghosh, professor cum a hardcore molecular biologist from Department of Microbiology, University of Calcutta. Professor Tania Das, ICMR Emeritus Scientist cum Molecular Oncologist from Division of Molecular Medicine, Bose Institute, Kolkata. Now we welcome Dr. Dilip Kumar Nandi, Professor in Department of Paramedical and Allied Health Sciences, and Dr. Shobhoshaji Pal, Associate Professor of Physics in Department of Pure and Applied Sciences of our college, and Professor Sandip Chattopadhyay, Head, Department of Biomedical Laboratory Science and Management, Vidyasagar University. All of them will chair different technical sessions in this conference. It will be a gross fault on my part if I do not mention here for our students that Dr. Suchismita Roy and Dr. Shravani Pradhan earned their PhD degree under the supervision of Professor Dilip Kumar Nandi only. Welcome, sir, to this conference. Finally, once again, I welcome all the resource persons, guests, teachers, staff, researchers, and students participating in this conference from this college and other institutes. I believe this conference will ensure a flow of knowledge as well as blessings from the experts to the novice. I wish you all a nice time in this college. Thank you very much.
Thank you so much, sir, for your enlightening address that so eloquently set the academic tone of this conference. We shall now proceed to the technical sessions. It is my privilege to invite Dr. Dilip Kumar Nandi, Professor, Department of Paramedical Allied Health Sciences, Midnapore City College, to come to the stage to chair the technical session 1, that is on microbial genome engineering strategies. Sir, please come to the stage. Now the stage is yours, sir.
I am very little person. The stage is big in contrast to my physical status or other than. Okay, very good morning everyone. And at first I deep sense of my gratitude to all of you for presence here. And I am very much proud about the presence of Professor Bhadutala Das. Have a big history behind Bhadutala and myself. And at first I gratitude to my founder director in this institution, and better to fathers of this institution and also the parents of this institution, and his love and his affection, everything in this institution from the since beginning to 2007 to till now. And another faculties, particularly Dr. Sudipto Chattopadhyay, Chakraborty, sorry, and obviously, initial professor joined here, Professor Subhusha Pal, and Dr. Suchismita Rai, Dr. Shravani Pradhan, and others. And after that, obviously, great Kunthal, dynamic and great Kunthal. But another faculties, very young, energetic faculties, and at the same times, very much energetic students, both the males and females. But I see here in front of the seats, not in equal presence in the according to the equality opportunity of the sex. And obviously, evergreen our Professor Nirmal Sarkar. He is very much vocabulary, and also the voice is very nice, and old but young energetics very present here. And obviously our program coordinators, and particularly Orpita, Abhishek, even also, please, sorry, don't mind, Surmila, right, and nicely managed in this occasion. Okay, and last not the least, and Kausik Das, my affectionate colleagues and students, best of the students and colleagues, and previous years, so many times I spent of the Kausik, and also Professor Sandeep Chattopadhyay. Since 1987, so far my memory concerns, from the Midnapore College to VIH and the Vidyasagar University, joined together with developed so many courses. And obviously today's program nicely organized last few years or few conference previously nicely, and I do not believe in the luck. I astonished that I have been present today, and after the suffering of acute disease myself. But may I request Professor Bhavatos Das, please come to the dais and share your views. Before that, you have sharing, Suchismita? The short CV of, no? Where is Suchismita? Okay. The Bhavatos Das, Professor Bhavatos Das, and not clear here. Professor Bhavatos Das is a distinguished microbiologist and microbiologist at the BRICS Translational Health Science and Technology Institute, Faridabad, and specialized human microbiome and also the antimicrobial resistance research. He holds the master degree in the life science and the biotech, that is the biotech and biochemical engineering, MTech, along with a PhD degree in the molecular biology. And so far my knowledge concern from the IICV, yeah, that time. And obviously PhD from the Jadavpur University. Okay. Next, following his doctoral degrees, postdoctoral degrees, research, focusing of the microbial genomes, dynamics, Professor Das at France or the Switzerland? France, yeah, France, maybe, France. Then the Professor Das leads the research lab that applies the cutting edge of the metagenomics and also the microbial genome engineering techniques to develop the next genome level biotherapeutics aimed at the testing of the metabolic and also the chronic inflammation diseases. His pioneering work Also the extents of the discovery and the development of the probiotics. Probiotics designed to promote the human health. As a recognized lead to the human microbiome research, Professor Das' innovative approaches are the driving new strategies in the development of the life biotherapeutics, offering promising solutions for the complex of the health challenges. His contributions continue to the shape the future of the microbiome science and its applications in the medicine. Currently, Professor Das' research focus on innovative microbial genome engineering strategies aimed at the understanding of the specific proteins and peptides and developing of the peptide-based drug, and also the target region of the human GI tract. His lab holds a diverse collection and also the microbial TASA associated with the gastroenteritis, sepsis, and both terms add of the patent bath and preterm bath. Thank you. Very short informations about the Professor Das. May I request Professor Das to deliver your lecture, better to share with your ideas, the new idea, innovative idea to present here, and our students and faculty will be benefited. Thank you.
Thank you, sir. It's a very extended introduction, so I think first time in my life. I just don't like it. And thanks, Kuntal, and thank you, all of you, for inviting me and giving me this opportunity. I completed my MTech in biotechnology and biochemical engineering from Indian Institute of Chemical, the Indian Institute of Technology, Kharagpur, and in 2003. And that times I visited Medinipur College as well as the Vidyasagar University several times, so I know this city very well. So after completing settled that my MTech, I was working on the microbial genomics in my PhD, and then also when I was doing my postdoc, I was trying to engineer the microbial genome in such a way that microbes could be used as a medicines. Okay, so that is the things I'm going to explain today, that the microbial medicines. We know that most of the medicine right now we prescribe, they are chemical entities, but you know there are several chemical entities, those are peptidal in nature, and this peptide is mostly synthesized by the microbes. So the idea is that if we can modify the genome of microbes, produce the same peptides, then it could be a microbial medicines. So briefly in my lab, so I have two major research components. So in one component we are working on the human microbiome, and the second in the antimicrobial resistance. And when I'm talking the human microbiome, so what we are trying to understand the different types of microbes that normally reside in our body. You know that the human body, almost each and every sites, those are environmentally exposed, colonized by the thousands of the microbial taxa. And those microbes are beneficial, and they do actually contribute to our human health and physiology. And sometimes what happens when we use the drugs, so these drugs actually kill the microbes. Okay. But microbe also try to, if it possible, I can also remove this one. Now it's okay. Can you hear me more clearly? Okay. So when we actually try to kill the microbes, so the microbe also try to survive. No? So that's why actually this antimicrobial resistance develop. So antimicrobial resistance, particularly in the commensal bacteria, is not a problem, but if that resistance acquired by the pathogenic bacteria, that creates the problem, particularly for the management of the infectious disease. So I'm not going to discuss today for antimicrobial resistance. So mostly I'm going to focus today my talk on the human microbiome part. So we have collected the sample, I can show that from the different parts of the India. So the major focus we have given to the gastrointestinal tract, the region, because the maximum number of microbes reside in the human gastrointestinal tract. Just imagine that if you compare the total number of somatic and germ cells we have, and if you look the number of microbes reside in the human body, you find that the more number of microbes reside in our body. So the first things we started to understand what is the microbes that normally reside in the healthy Indians. Okay, so people living in the urban area, rural area, hilly, and even to the Andaman Nicobar Island. So we collected their fecal sample to understand what is the normal microbiome of the healthy Indians. Then we try to understand how those microbes differ when people actually suffer different types of health disorder, that including that severe acute malnutrition, type 2 diabetes, pre-diabetes, non-alcoholic fatty liver disease, the pneumonia, inflammatory bowel disease, gallbladder cancer, and recently also we started on the sepsis. Okay. So if you simply compare, you always find there is a deviation. So there are two reasons. These deviations may be because whenever we have a disease, we have been treated with the drugs, the drug can change the microbiome composition. And second possibility, those deviated microbes are actually the pathogen that induce the disease. Other than the gastrointestinal tract microbes, what actually we also working right now to understand the reproductive tract microbiome of the Indian woman. And I will explain that this is one of the largest cohort right now we have in the Asia, where almost like 13,000 pregnant women are enrolled. We collected their high vaginal swab samples and then try to understand how actually the microbes reside in the reproductive tract, influence the birth outcome, whether a woman will give the term birth outcome, that is after 37 weeks of gestation periods, or whether a woman deliver preterm birth before 37 weeks of the gestation periods. So that can also be influenced by the microbes reside in the reproductive tract. So that we are trying. Even also we explored a small study to understand the breast milk microbes. So even the woman breast milk is not sterile. There are a lot of microbes reside. Actually those microbes are the initial seeds in the neonates to get colonized and later on develops. And also we started working on the organ microbes. So now we have the different types of organ that includes like tissue sample from the liver, then stomachs, gallbladder, then respiratory tract. So wherever people come for the surgery, we try to get those biopsy samples and understand whether any microbial invasions and that is associated with the disease. So in overall the last I can say the 10 years what is currently our understanding about the human microbiome. If you look to this slide, that all the sites we have the microbes, but the microbial load differ depending on the body sites. The highest number of microbes reside into the human clone. You can find that almost like 10 to the power 11 per ml of the samples you have the microbes. And the minimal number of microbial load we observe in the stomach, because stomach is highly acidic. In this condition, microbes cannot sustain. Only very few bacteria, those are resistant to acid, they can sustain. Otherwise, you have all the others. You can reproductive tract, you have the breast milk, every organs, you have the microbes. Now, what types of microbes? Whether microbes reside in all the sites, they are similar or they are different. So this is the one study books that we published 2022. What we observed that each body site there is a keystone bacterial structure. That means a specific bacterial structure dominated each of these body sites. If you look to the reproductive tract, this is dominated by the lactobacillus. If you look to the gastrointestinal tract, particularly for the colon, this is dominated by the different anaerobic bacteria that include Bhadutala, bacteroids, Rubinococcus. These are all the stick anaerobes normally reside into the colons. Skin is mostly dominated by the different types of Streptococcus, Propionibacterium, Actinobacteria. Similarly, if you look to the lungs, it also be dominated by the different aerobic bacteria. So depending on your body site, you have different types of microbes, and each types of microbes basically produce some metabolites. Those are very important for our health. If you look to the gut, they produce short chain fatty acid. Those are basically signaling molecule. They actually modulate your lot of behavior and health physiology. Same thing if you look to the reproductive tract, they produce lactic acid, acetate, propionate. So those are also important to decolonize the resistance bug. So you have different types of bacteria, and they do also produce different types of the metabolites. Those are very important. So this is the normal human gut microbiome. But when we go for the disease context, what we observe that nowadays, almost each and every organ, there is an association with microbial dysbiosis. So when I say the microbial dysbiosis, that means if you compare the microbiome composition and function of the healthy human versus the people suffering a specific disease, there is a substantial differences that we call the dysbiosis. The two signature that what we observe that metabolites, those are very important for our physiology, including short chain fatty acids, polyphenols, dopamine. So their level is reduced, and the other metabolites, those are very important for inflammations like trimethylamine, deoxycholic acid, LPS. Their number is increased. So there is basically differences in the metabolites that present locally or in our systemic circulation, and you can say easily those are associated with the different types of disease. And these are the different articles, if you go, that we published in last 10 years, almost each and every study, normal microbiome of the people living in urban and rural areas. We had also completed several international studies where we actually compare the microbiome composition of the healthy peoples living in India versus Japan, living in India versus Danish, and these are all published literature. So in one slide, so if I just want to explain how actually those microbes are associated with the disease, I can give this example that whenever you have a specific types of disease, you do find that either some of the microbial taxa, their number is increased, and some of the microbes, the number is reduced. Microbes, those are commensal in nature that provide benefits, their number is reduced. And microbes, those are pro-inflammatory or opportunistic pathogens, so their number is increased. And few microbes, those are actually common for different diseases. For example, if you look to the cholincellular patients, you do find that their number is increased of people suffering in non-alcoholic fatty liver disease, as well as the people who is suffering in type 2 diabetes. Same thing if you look to the commensal bacteria, Faecalibacterium prausnitzii, their number is reduced in malnourished conditions, as well as their number is reduced in the ulcerative colitis. So you have disease-specific microbial signatures, as well as you have the microbes, those are common across the disease. So this is the common finding in one slide. So now the hypothesis that we made, we know that there are normal microbes and there is a deviation of the microbes in the disease context. Whether we can use the beneficial microbes that lose dominance or are depleted during the disease can be used to develop a therapeutic candidates. Because these microbes produce some metabolites. Those metabolites are important to maintain your physiology. So if you can use a consortia of the bacteria, or if you can modify their genome, so there is a possibility those microbes itself can act as a therapeutic. So I'm giving few examples that actually we have few success story where we are able to develop a microbial consortia, synthetic consortia, and ultimately we transferred those consortia to the industry. The first one I can give example, the study that I explained, the reproductive tract microbiome. So almost like 12,370 women enrolled in this Garvene cohort. Those women actually screened longitudinally. So they enroll in this cohort before 14 weeks of their pregnancy. Then we collected second sample in 18 to 20 weeks of their pregnancy, and the third sample 26 to 28 weeks of their pregnancy. Now, when you are looking the microbes, there are microbes, they are auxotroph, the microbes, they need specific nutrient, we don't know. So there are so many microbes, you cannot culture them in the laboratory conditions. Then how do you know what types of microbes resides? For that, what we did the culture independent analysis. That means we collected the high vaginal swab samples, extracted all the DNA, and then we did the metagenomic analysis. So metagenomic analysis, either by targeted metagenomics to amplify the 16S or go for the complete analysis. So that will give you the idea what types of microbes reside. Now once you know that what types of microbes and what is their status of the auxotrophy, accordingly you can design the growth condition and you can isolate. So once you know that almost close to 31 or 32 different lactobacillus species normally reside in the women reproductive tract, those are important for the health, we actually isolated all of them. And close to 350 microbial isolates we had. We have done all their genome sequencing analysis, and what we observed that there are two keystone taxa. One is the Lactobacillus crispetus and second one is the Lactobacillus paragaceri. Those have the therapeutic potency. So whenever you have the Lactobacillus crispetus or Lactobacillus paragaceri in the reproductive tract of the women, they are less susceptible for any types of microbial infections. They are also giving the term birth outcome. So that's why we feel that most possibly they have some function that provide these protections. And this is very well known that lactobacillus produce different types of peptides. Those are antimicrobial in nature. They reduce the growth of the reproductive tract pathogens, and that could be the one of the reasons. So we have grown this bacteria, and then we actually took the cell-free culture supernatant to understand their pathogen resistance function. What we observe that these specific sets of the bacteria, they completely inhibit the growth of the Gardnerella vaginalis, Klebsiella pneumoniae, E. coli. They are the normal microbes, reproductive tract pathogens in the Indian scenario. In addition to the pathogen resistance, what we also observe that they do also have anti-steatotic activity. That means they reduce the fat accumulation in the liver as well as fat accumulation into the viscera, and also they produce the anti-inflammatory substance. So that we change. Then we make the consortia. It's not only one bacteria. So you have a set of bacteria. If you're making a synthetic consortia, then you have the therapeutic potential. So we make two consortiums. One consortium that based on the multiple strain of Lactobacillus crispetus, and the second consortium that based on the Lactobacillus paragaceri. Now those Lactobacillus consortiums been tested in the preclinical animal model, and we transfer it to the industry. Okay, now almost like 21 countries nowadays across the globe, they're basically selling both the product. So how actually we came to conclusion that this is the consortia, they have the therapeutic potency. So you need to understand that story. So first thing what we did in the slide number 1, I can explain that we selected initially 40 Indian women to understand the normal reproductive tract microbiomes. Okay, we did the metagenomic analysis, and this is our finding that this is particularly the Smithia, Gardnerella, and Megaspora dominated. They basically are prone to the preterm birth baby. That means the woman can deliver their baby before 37 weeks of the gestation period. So this we done around 116 samples collected longitudinally from 40 women. Since the sample size was low, we want to validate that this is the correct. Now here we increase the sample. We have almost like 600 sample now collected from 200 pregnant women. So 140 those are from the term birth, and 60 of them are the preterm birth. And we exactly identified that what we had published previously. We can reproduce the same phenotype. These Lactobacillus species are associated with term birth outcome, and the Lactobacillus inerts, Smithia, and other non-Lactobacillus are associated with preterm birth. Now this is one of the strong biomarker. You can screen the woman to predict the risks whether a woman will deliver later on term and preterm. But sequencing is a very expensive experiment, no? And we do not have these types of facility everywhere. That's why we develop another one method, a district-based method. Very quickly you can detect the presence and absence of those 8 or 9 different bacteria. Okay, this is currently now in the development. So we receive recent fund from the Tata to make these dipsticks to available even in the primary care hospitals. So this is all the Lactobacillus since we had. So what we did, we just first try to understand the Lactobacillus that we isolated in India. whether they are unique if you compare with the lactobacillus that isolated across the globes. Okay, around 790 lactobacillus genome we compare, and we do find that there are several lactobacillus isolated from the Indian women. They have unique genomic signature. Most of the genomic signature are associated with the fast resistance or sometime of the MP productions. So here this example that when we started with the commercialization of those lactobacillus, those are very much unique to India. So we look to the Indian market. So what types of different product is available? You could find that there are multiple products. Those are mostly by the different pieces of lactobacillus, all mixed of the lactobacillus available in the Indian market. And it was very unfortunate that not a single lactobacillus is isolated from the India. Each and every lactobacillus or other bacteria we use in India, we are importing from the outside, either from Europe or US. What is the problem? Same bacterial species we do have the better bacteria. Instead of using our own product, we are using something coming from Japan, Italy, or from the US. So that is the idea that even the Indian company, there's a big company, the Sanjayota Newmandis, they are one of the largest manufacturer, produce this seed, and they basically sell these lactobacillus across the globe. All the lactobacillus you could find that they're coming from the outside. Then we had a discussions with the deputy secretary, and also they asked all these vendors to come. So what is the issue? Why don't you use the Indian isolates? The first thing came, that is the safety. So safety is the most important for developing any therapeutics, because you are going to use it for the human. Whether this is effective, whether this is good, that is the secondary. So safety is the primary. So we do not have substantial data from the Indian isolate that those bugs are safe for human use. That is one of the major concerns. Second concern, also in India you need at least 5 different levels to the regulatory approvals. Most of the people work, they have 2 or 3 regulatory approval, rather regulatory approval are not there. Even for the bacteria, you need the NBA, National Biodiversity Act, their approval if you go for the commercialization. Since we had very good support from the department, so we able to manage all types of regulatory approvals. We have done all the complete the safety profiling study. And now this same company, now they have given almost 1 share for each of these consortia taken from our laboratory, and now they are selling across the globe. So it's not like that the company are not interested, so but they also always need this data need to be very complete. So this is the indigenous strain, in-house strains I was explaining that we have done the analysis part, and we did this, and how these bacteria we identified. So when we did that, the complete genome sequencing analysis, we observed that they produce multiple types of the peptides. Those are antimicrobial in nature, and they give actually complete protection against different types of intermediaries. So we have tested initially with the E. coli, Klebsiella pneumoniae, Gardnerella vaginalis, and then also we have identified what functions of those bacteria provide this antimicrobial activity. We have taken the cell-free culture supernatant, did this mass spectrometry, and we do identified those peptides, those are responsible for this activity. And then we have also tested against the commensal bacteria, because if this peptide is killing everything, that is also not good. So if you look at the antimicrobial activity, you find that those peptides are so selective, they work against mostly gram-negative pathogens, but they don't work, those are the commensal. So if you look to the lactobacillus different species, they don't have activity. And the antimicrobial activity we find because of the 2 different functions. One is basically the lactic acid produce this bacteria, the yellow color. In addition, this bacteria produce the peptide, that is the green color. So either the antimicrobial peptides or the lactic acid produced by the lactobacillus gasseri and lactobacillus paragasseri, they have this production activity. So we have tested, and we have also published this study. We also make sure that these lactobacillus need to survive in the gastric acidity condition, because they need to pass when you are using through the oral routes, and also they need to be resistant against the bile salt. We have also tested bile salt, and there is no reductions of the bile cell activity. This is another interesting the story came from the same bacteria. It's not only the protection against the pathogen, But what we observe that this bacteria produce some molecules that actually also reduce the body weight, particularly they reduce the steatosis. So we did this experiment with the animal, and you could find that if the animals fed with the high fat diet, their body weight is increased over the time. But if you are giving the animals with the lactobacillus gasseri, you find that their body weight is significantly reduced. Okay, and then we have also tested with the other activity that is associated with this your body weight, that is the blood glucose level and also to the insulin level. So this is the study. We found we get that the liver samples, and you can find even from the liver sample, this is very clear that when you have only the CDHF and when you have the CDHF plus lactobacillus gasseri, there is a strong improvement. So they are important because they produce some metabolites, and the recent study also came that they produce a very small molecule. Those are basically anti-inflammatory in nature. And the final things that we have done, that once that you produce that lactobacillus, if you want to commercialize, those microbes need to be viable in the room temperature for the longer times. We have also tested the viability of the lyophilase. You have to just address these matrix that you are going to use, because that matrix need to be safe for the human, and that matrix also need to support the growth of this bacteria or viability of the bacteria for the long time. So we use mostly the sucrose and the gelatin, or a combination of the sucrose and gelatin, and we tested till 98 days. You could find that even if you are keeping them at room temperature 4 degree or -20, most of them are viable. So this is the things we product. So this is the story of the lactobacillus that we actually transferred to the Sanjayota, and this is a synthetic consortia of the wild type bacteria. So you identify what bacteria, their number is depleted in the disease conditions, and then test that if you are giving these microbes as a supplement, whether they can improve your physiology or not. So this is one. And the second thing, now we are doing, and we are in very advanced stage, the Gates Foundation is coming to support us, that whether we can use microbes directly as a medicines. Okay, that is the hypothesis that the peptide therapeutics hold an enormous position. You know that nowadays everybody want to slim, and because that everybody is going toward the GLP-1. So GLP-1 is a drug of wonder that reduce obesity, and it's a basically hormone produced by the human body, just only 31 amino acid long hormone. So question is that if it's a peptides, and if the bacteria produce this peptide in your gastrointestinal tract, whether they will serve as a medicines or not. So that's why you need to modify. And the another beauty of that, if you able to engineer the genome of bacteria to produce this peptide, you can also deliver those peptide in organ specific manner. There are different bacteria, they colonize into the gut. If you want to deliver that peptide into the gut, better to go for the bacteria that reside in the gut. The bacteria that present in the reproductive tract, you can select. So if you are selecting your microbial chassis, you can also deliver your peptide in an organ specific manner. So that is the two things we started working. So what types of peptides we want to select, and where do you want to deliver that peptide? Based on that, you can select the microbial chassis. So this is the idea, but problem is that whenever you are going to use a recombinant microbes, there are a lot of regulations or the regulatory hurdles that you need to fulfill. So FDA has published in 2019 that if you want to use recombinant microbes as a biotherapeutics, what would be regulatory criteria you need to fulfill. First thing, that the manipulation that you are making, that need to be very stable. It should not lose quickly. Second, the same function should not transfer to the other bacteria, that can create a problem. And there must not have any antimicrobial resistance functions, because later on if you simply want to remove the bacteria, you should be able to use any antibiotic to remove. So we started working. So we first developed a indigenous genetic tool that we patented. So this tool have the capacity to go and integrate into the microbial genomes. So once they integrate into the microbial genome, we have the 3 different types of enzyme substrates that can remove the construct and that can keep only the gene that is responsible for synthesis of this peptide. So we manipulated into the chromosome, so that's why they are very stable. We remove the plasmid after manipulation, so there is no more antibiotic resistance gene, or there is no more plasmid backbone. So this is a complete safe recombinant bacteria. Those are antimicrobial resistance free. And the bacteria that we use, that is the recombinant Escherichia coli Nisel 1917, they are already in the human use. This is one of the most well-known probiotic bacteria people are using almost like last 100 years. So this is the different peptides initially we selected. So we selected the human encoded this GLP-1. We make a multiple combination of the GLP-1. And why GLP-1? Because GLP-1 is now, say, a wonder drug for the metabolic disease. People's been used for type 2 diabetes, for obesity, and recently also for the non-alcoholic fatty liver disease. And this is a very small peptide. It's like 37 amino acid long peptides normally produced by the human intestinal cell. So if you can use a microbes and colonize those microbes into the intestine and you can produce, then there's a diet. So that's why we did. But GLP-1, their half-life is very small. So okay, within like 5 to 10 minutes, the human enzymes can also cleave them. So that's why what we also did, we make different receptor agonists, that is the mutated versions of GLP-1, where the half-life basically get extended. So that includes, there are already reported GLP-1 receptor agonists, that is exenatides, liraglutides. So we use all those resistant genes to engineer the genome of E. coli Nisels, and then we test in the animal model. So this is the simple things what we did. We selected those peptides, those are known, and the few peptides that we identified first time into the gut microbiota, engineer the genomes of the E. coli Nisels in such a way that this peptide will produce. Once this peptide will produce, they will secrete out from the cells to the extracellular phase, and then it should work. So we use mice high-fat diet model, mice that actually supplemented with these recombinant E. coli Nisels, been fed with the high-fat diet, and then we have tested with your insulin, glucose, other enzymes, cytokine levels, liver weight, body weight, and everything. And here we have done. So the first thing, the manipulation part when we did, so we confirm first the integrations through the PCR, then we did the complete sequencing to understand exactly where this integration happened. Since those microbes reside into the human gut, so human gut is completely anaerobic in nature. So you need to make sure that your promoter is not sensitive to oxygens. That's why we measure their expression both in aerobic and anaerobic conditions. And finally, you need to show that those peptide is coming out from the bacteria and they're intact, so that we confirm by the mass spectrometry. So we have done the complete profiling that is as per the requirement of the FDA. Now, this is the different types of sample. I explained that. So we use first the pure peptides as our positive controls, then use the entire microbes, lyse those microbes, measure the GLP level in the cell lysate. Since this peptides is supposed to secrete out, we have also taken the cell-free culture supernatant and also measure that peptide. And finally, you need to ensure that once you are supplementing this bacteria, you should be able to detect these peptides into the blood of the mice. Now here we face another problem. If you compare the GLP-1 sequence of the human and mice, they are 100% identical. So how do you know that the peptide is coming from your bacteria or this is the indigenous peptides? That's why what we did, we also introduce specific mutations. Okay, now we have changed one amino acid just to make sure that the peptide that is produced by the recombinant E. coli is coming to the blood. And we able to detect all of them by the mass spectrometry. And when you look to the level of the peptides, it also reflects the numbers of microbes that actually present in your gut. When you give the E. coli Nisel 1917, what we observe that the maximum number of CFU come at 48 hours, okay, and by 72 hours it completely flushes out. So you could also find that in the time period, so 48 hours you have the highest number of peptides you can detect into the blood, and over the time again these peptides are not detectable. So this is the different types of peptides that we detected. So we detected with pure peptide, with the charmins, then we have detected with the bacterial derived peptides, that is this is the Parabacteria distationis, and also we have the mutated GLP-1, and sometimes we have given the wild type bacteria from the gut, and you can also detect this apeptide. So all the cases you have detected, and each of the peptide fragments through the mass spectrometry, those are coming quite viable into bacterial cells. And finally we tested in the mice model. Now what we tested, this peptide, even if you are able to detect into the blood, they need to be functional. So what is the function of this peptide? This peptide actually induce the secretion of insulin in the presence of the glucose. That's why what we have done, we measured first the fasting blood glucose level. If you look to the blood glucose level, semaglutide is the standard peptidal drugs that normally people use. We use that as a controls, and then we challenge this mice with the glucose and then measure the insulin level. You could find that this is the control, negative control, where mice is fed with only PBS. Then we have given the mice with only E. coli cells. So E. coli cells that not have this peptide, and then we have given E. coli cells that can produce the peptide, and this is the standard drug available into the market, that is the semaglutide. And you could find that once you give this specific bacteria that has the capacity to produce this peptide, they substantially increase the insulin level in the presence of the glucose. So now we also proved that this actually recombinant bacteria not only produce that peptide produce this bacteria, this is also compatible with the human physiology and they are functionals. So I can say that this is the pipeline that we follow till now. So we started with selecting the peptides. So currently not only GLP-1, so we have 13 different peptides able to modify the genome of the bacteria that include different types of GLP-1. Other than GLP-1, IL-10, there is a huge market value. A lot of the inflammatory disease people use IL-10. And also we started working with the insulin, because you don't need to take the insulin each and every day. So those peptides we have basically engineered with different types of bacterial chassis that include E. coli, BC, and lacto. And then we actually measure this level of this peptide in the genomic manipulations, tested with the animal model. In animal model, particularly GLP-1 and GLP-1 receptor agonist been completed. We have also started working with the IL-10 and insulin into the animal model, animal that fed with either normal chow or IPAT high fructose diet. In the normal chow, what we also observe that even if you give this bacteria that produce the GLP-1 receptor agonist till 24 hours, there is a very minimal increase of the body weight. Once we make that recombinant bacteria, the next step what we also ensured that we able to produce this recombinant bacteria in the at least medium scale volume, so 5 liter to 10 liter scale. In the bioreactor we have also produced. Then we also lyophilized this product, and we have tested their viability in the different temperature that include room temperature 4 degree and -20. And currently we are proposed 2 clinical trials with all India Institute of Medical Science, one particularly for the obesity, and second one we are working for the recurrent urinary tract infection, and we are also now in discussion with this product with the industry. So in summary, I can say that whenever you want to make a microbial biotherapeutics, first steps you need to know what are the commercially successful probiotic that should give you the information what could be the best microbial chassis, and what the most important parameters that you need to concentrate. The microbes that you are going to select, they need to be minimum sensitive to atmospheric oxygen, because ultimately you have to produce this bacteria. After production you need to sell this bacteria. You cannot maintain anaerobic condition into the market, so they need to be minimum sensitive to oxygens, and they should have minimal oxotropy, because if those microbes are oxotrope, then you need to give the supplement that will increase the cost. Then also they need to survive in the different condition, particularly the freeze drying conditions, because you have to lyophilize them. If the microbe grow very fast, then it always good for the industry, because that will reduce your production cost, and also your microbes need to be high cell density. But this is the ideal case. So it may not be possible for all the good microbes to achieve each and everything. In that cases, what you can do, you can actually find that there are some functions, those are important, and if you can manipulate them. That's why we started that Faecalibacterium prausnitzii, they produce IL-10 like peptide, but Faecalibacterium prausnitzii you cannot manufacture in large scale in anaerobic condition. We take the entire biosynthetic gene cluster and then we produce the recombinant live biotherapeutics. They have the similar types of functions. So that is the roots. This is my collaborators that I work. I really like to thank first Professor Kyosu Takida, the first study that we did the Indo-Japan, then Rupak with the Indo-US and FX in France, my postdoc boss. These are my national collaborator, this is my institutional collaborator, these are my funding agency, and this is my groups currently I am working with. So most of the lactobacillus work I explained today done by the Lakshmi and the Proxima, and the recombinant work is doing by my PhD student Bharti Kajal Tansi. And thank you for your attention. We'll be very happy to take your questions.
Thank you very much for your innovative, even also the illuminative presentation, and obviously informative in the field of bacteriotherapy, particularly in the bacteriotherapy. And it is very much needed to in our society. But doctors are not acceptable in our society easily. This is the most lack of behind this, but gradually increase after the 1980s and onwards. The prebiotics and probiotics are gradually accepted and increased in our society, used by the doctors and the other society, other people. And may I request all the students, please settle down. Settle down. Please sit down. Hello, any problem? Please sit down. Any question from the students? Any question from the students? No questions? Yeah, obviously. And next, the research scholar. Actually, so many researchers are working here regarding the micro. You may. কি চাইছো তুমি কে বলবে No researcher. Now the evergreen persons, Professor Nirmal Sarkar. Any questions?
Sir, thank you very much for your illuminating but fascinating lecture. I have got some simple questions. First thing, you showed in one slide that 5 bacterial species are causing preterm birth in human women.
Yes.
Should all these 5 species exist together, or a single species can also cause preterm birth? This is my first thing that I want to know.
Okay.
And the second thing, do you think that it is possible that these bacteria are somehow adversely influencing progesterone level or other hormone levels in human women? And finally, when you are trying to make therapeutic use of genetically manipulated bacteria, don't you think, or how can you ensure that in course of time the animal body or human body will not develop any humoral immunity against the bacteria that you are administering in animal body or human body?
So very nice question, sir. The first questions, so whether a single microbes or a group of microbes that associated with preterm birth baby. So we observe that depending on the microbial species. So when you have particularly the Gardnerella vaginalis, even only presence of Gardnerella vaginalis sometimes is sufficient to induce the preterm birth. Okay, but this is not case for all the others. So sometimes, like if you look to the E. coli, so they also basically induce or in the risks of preterm maximum when you have the coexistence of other bacteria. Okay, and what current hypothesis we have and some of the preliminary data, this bacteria actually induce the matrix metalloprotease. Okay, and because of that, the quiescent stage is basically over and they enter to the active phase, and they go for the bacteria. Progesterone level we also measure, but we do not find any differences in progesterone level based on the microbiome composition in the gastrointestinal tract. But matrix metalloprotease level is significantly different, and there are studies been also reported. And your last question regarding the development of resistance of the human body. So I feel that that possibility is minimal, because the peptides that we selected, that is the native peptide of the human. If you look to the GLP-1, so GLP-1 is produced by the human body, and there is no development. Same thing if you look to the insulin. Insulin is basically produced by our own cells. So that's why if you are giving the same peptides that human body is produced, there might be a possibility.
But sir, if the woman is diabetic, having type 1 diabetes and not producing insulin?
Oh, that's why they are giving. So when we are diabetic, we take that insulin, because there is a deficiency of insulin. Okay. Anything, any drugs you take in excess amount, definitely there is a possibility of developing a resistance. So how preciously you are using your medicine. Same thing for the antibiotics. You use antibiotic precisely, you find that minimal rate of emergence of resistance. But if the use is normal, then it's going to be issue. Insulin also, it's happened over the time, over the age, body may develop resistance. There's always possibility.
Thank you, sir.
Professor Sandeep.
Thank you, Professor Das, for your nice lecture, because this is the demand of the days, I think. And one thing is very interesting, your institutional collaborator, Dr. Jayanta Bhattacharya.
Yeah.
He is my very close friend, my batchmate, classmate, and from the childhood actually we studied together. So anyway, so I have some basic query about that. Now you are talking about the different types of probiotics. Actually these are all coming from the foreign countries.
Yeah.
Okay, how much these are favorable for our gut environment to survive? So that is a big question of the day, because nowadays whatever the probiotics you will find out in the market, so maximum probiotics are coming from there. That is very true.
Yeah.
So that is my first question. And then another thing is that nowadays doctors, they are not suggesting this type of lactobacillus group of probiotics. Particularly I am observing in my town. So if you go to the doctor, immediately they are suggesting you some fungal probiotics, particularly the Saccharomyces boulardii. So in this regard, if you have some opinion, why they are doing so, what is the possibilities of this type of fungal probiotics, and how the probiotics are surviving, those who are which are coming from, because we are making our gut in such a way that we are taking the yogurt, that is commercial yogurt, homemade yogurt we are not using. So the total gut environment is being changed. So in these perspectives, how these microbes are working, and my second question is regarding the use of fungal probiotic.
Thank you, sir. So your first question, particularly whether the microbes that is coming from outside will work with us or not, most possibly not. So I am working currently with Get Foundations in a project where we are making a consortia that been isolated from South Africa, then US, Europe. And when they try to use this consortia to a population in Bangladesh, they find that there is a very minimal effect. They try to understand that this is the same Lactobacillus crispatus present in the European women, also present in the Bangladesh women, why they are not working. So what we observe from our study, that most of these bacteria, they are quite susceptible to phages. We have different types of bacteriophages that actually kill this bacteria. But when you are taking a bacteria from the native population, these phages, through their CRISPR-Cas system, they acquire resistance against the bacteriophages. That's why if we are giving an indigenous isolate, since they are resistant against that specific bacteriophages, they get colonized. But if you are coming something from the outside, since they do not have that resistance against the phages, they don't work. So it's always better to have your probiotic from the indigenous populations. Okay, that is the first question. Second question for your fungi. So if you look to the Indian population, 94% of the healthy Indians, they have candida in their gut. We don't have Saccharomyces in the Indian population. So Saccharomyces is the native microbiota of the developed country. So if you look our Indo-Japan and Indo-Denmark study, where we analyze both bacteria and fungi, what we observe that Japanese people, they have Saccharomyces, European people, they have the Saccharomyces, but the Indian people, we have the Candida. So the possibility of having health benefit from Saccharomyces is very minimal, because that is not native to our microbes. So whenever you are looking the gut microbe, you have two types of interactions. Okay, one is the competitive, another one is the synergistics. So Candida has the synergistic interactions with the Prevetola. Prevetola is our normal bacteria, and Saccharomyces has the synergistic interactions with bacteroids. Bacteroids is the native microbiota of the developed people. So that's why Saccharomyces work over there. Nobody look on that. Since it's work in Europe or Japan, they do also prescribe to the India. But if you actually analyze the gut microbiome, you could hardly find that those Saccharomyces ever even get colonized into the Indian gut populations.
Thank you, Professor Das. Very nicely answered the questions, particularly queries. My Lucy question is, and what are the, you have developed any types of the industrial product regarding the probiotics for preventive of the lung function? But so minimum quantity of the microbiome presence in the lungs, as I am suffering from many times in the lungs problem. That's why my query of this.
No, sir, lung probiotic we have not developed, but what we did, the microbiota of lung when people were suffering this SARS-CoV-2. So we published 2 articles. What we observed that some of the people who develop severity after infection with SARS-CoV-2 and people who has not developed any clinical symptom, what is the differences in lung microbiota. So what here we also observe that the co-infections with masked adenoma virus and presence of Klebsiella always induced towards severity. And if the people, they do not have the masked adenoma and Klebsiella, most of the time they are asymptomatic or they have the minimum severity. That is the observation.
Very good. VLC is your product? VLC3? No, this is the industry will be product, but very very effective product in the available in the market in contrast to the other probiotic. As because so many probiotic are available in our country, they are producing by the so many industry, particularly pharmaceutical industry, but not equal weightage of the microbiota presence in each of the probiotics. This is the most problem in our society also. Okay, it is a big problem and will be solved slowly. Thank you everyone, and I think enjoy everyone. Please shake your hands for his queries and the answers nicely. Thank you. Please come.
Thank you so much, sir. Thank you, our esteemed speaker, for this wonderful technical session 1. Now, I would like to request our research scholar to felicitate Dr. Sagarmoy Ghosh, Professor, Department of Microbiology, University of Calcutta, as a mark of respect and gratitude. Sir, please come to the stage. Now, sir, please take your seat off stage. Now, we would like to move towards our second technical session of the day. I would like to request Dr. Sandeep Chattopadhyay, Professor and HOD Department of Biomedical Laboratory Science and Management, Vidyasagar University, Midnapore, to come to the stage to chair the technical session 2, that is on synthetic biology in disease management. Dr. Chatterjee's research area are related to molecular and cellular and environmental toxicology, reproductive endocrinology and immunology, physiology, pharmacology since last 27 years. He has about 20 years of teaching experiences. He was postdoctoral fellow in Oklahoma State University, USA, and also he has received awards at Chitralekha Mukherjee Memorial Oration Award 2023, CRS Silver Award 2025, FNESA in 2025. 14 research scholars completed their PhD under the guidance of Dr. Chatterjee, and currently 6 research scholars are working under his supervision. Dr. Chatterjee has 57 publications in reputed peer-reviewed journal with high impact factor. His citation index is 1121. He has completed 5 research projects funded by national and international organizations. Now, the stage is yours, sir. Please.
Very good noon to everybody. Actually, first, I am very much honored that Midnapore City College has given me the opportunity to chair a session. For that, actually, I am very much thankful to the director, Professor Pradip Ghosh, our principal of this college, Sudipto, then our vice principal here, and other all dignitaries. Those who are present here, Saboni is there, Suchismita is there. Repeatedly they are calling me that, sir, you have to come. Okay, then no problem. I have holiday today. Okay, so my university picnic is going on. So I actually I skipped that. Actually, that is generally celebrating like a festival. So this time I skipped that picnic. So I think that it is better to come here to get some experience regarding the recent knowledge on the synthetic biology and sustainable development. So initially when we have started our journey, actually for the research, particularly those who are doing their research work here, so generally the people, most of the people, they are running after the hypothesis. So anyway, that if you have some hypothesis in your mind, you have to prove that hypothesis by any means. Suppose when you are doing your research, you are doing your experiment, you may get some awkward results, actually, which is not generally favoring your hypothesis. So immediately some kind of manipulations or this type of fabrications of research that is doing, because this is not actually, particularly when you are thinking about this type of synthetic biology and other things, actually you don't have such type of ornamentations or fabrications of research. So ethics is a very big part, because actually all the students or research worker, you should know that what is the significance of maintaining the research ethics. anyway, that you can do so many things. That earlier, in the earlier lecture, you have seen that how we can take the probiotics from the vaginal surface of the pregnant mothers. And then from there we can develop so many probiotics commercially, and so that we can also reduce the risk factors of the different types of disease like NFLD and other things. Every day people are suffering from that, because in our country there is a significant percentage of populations are suffering from NFLD. If you go to the USG lab, generally most of the people, they will find out that grade 1 fatty liver disease initially. That is very common in Indian populations. So that is the necessity of this type of synthetic biology research, and so that not only that we can do so many things from this type of research. So I think I don't want to waste this technical session by giving you some advice. So rather than, it is the time to welcome our next distinguished speaker, Professor Sagarmoy Ghosh. Actually, his lecture is today the A hepatitis B virus pre-genomic RNA-based mini-gene transcript utilizes polypyrimidine tract binding protein regulated NXF1, NXT1 nuclear export pathway. So before going to the mainstream of the delivery of his lecture, I would like to introduce Professor Sagarmoy Ghosh. Actually, he is the professor of Department of Microbiology, University of Calcutta. And Professor Ghosh is currently working as a in Calcutta University, and he was completed his BSc degree in chemistry from University of Calcutta, and then he shifted towards biochemistry in his postgraduate curriculum in 1992. So after that he was working at the Indian Institute of Chemical Biology, Kolkata, and there he completed his PhD in 1998. And generally his research work is involved to protein structure, to analyze the protein structure and function. Dr. Ghosh completed his postdoctoral research work from the Department of Genetics, Duke University Medical Center, Durham, Newcastle, USA in 1998 to 2001. And his research specialization was related to coupling between transcription and splicing. He was working as a CSIR Senior Research Associate, Manovikas Biomedical Research Center between January 2002 to July 2003, and then joined as an assistant professor of biomedical professor in the Department of Microbiology, Calcutta. So currently he is working in the area of RNA maturation pathways of hepatitis B virus and small RNA mediated gene regulation in bacteria. He has completed several projects funded by different governmental organizations of India, and 6 research scholars already awarded PhD under his supervision, and many research scholars are still working in his laboratory. He has published his research work in many reputed national and international journals and books. Now I would like to welcome Professor Ghosh. Now dais is yours. We are very much awaiting for your talk.
Um, Thank you very much for such a kind introduction. And I'm absolutely, so to say, delighted to come over here in Medinipur City College. For this is my first time here. And I believe, the science, the education of science and technology that in our state of West Bengal has really been kind of spearheaded by people from all over the state, of course, but especially in Midnapore. And as I come from the background of chemistry, I have got so many friends who came from this particular area. And in every field that you go, in India, of course, and abroad, the Bengalis that people go, when they go abroad, you will find most of the people have a background or some kind of root in Midnapore. So I'm pretty sure, and of course, I often have to come to Vidyasagar University Microbiology Department. I see the enthusiasm and spirit of bringing forward the culture of science in all aspects. So I only have high hopes, and I'm sure in near future we'll see many talents coming out from this area and enrich us in many different ways. But without much ado, I must continue what I'm here to talk about. So mostly my work will revolves around RNA synthesis and how it gets matured. And there are two wings in the lab, one that works on the bacterial RNA, bacterial small RNA mediated regulation, and the other one is the viral RNA and how it is being controlled. So today here I'm going to present to you the work that I've been doing for the last say 15-20 years on hepatitis B virus. Disclaimer first, we do not really work with the living virus particles because there are some ethical and biosafety issues to work with hepatitis B virus unless you have a certain facilities, and working in the university environment doesn't really is always the right place to work with live viruses like that. So we make a shortcut. So the shortcut that we make is that we actually clone certain regions of hepatitis virus and express the RNA from there and work on that. So slowly we'll go there. So what we'll be discussing here today. So first of all, we work on a mini gene. That's what I was talking about. That we have made. We generally make a mini gene transcript. So this is not going to make any virus. So we are going to look at the maturation of this mini gene transcript, and then we are going to look forward to the ways through which this particular mini gene can exit the nucleus. Now, before going to the details, a little understanding of hepatitis B virus biology would be essential. This is what is happening. The HBV variant contains a partially double-stranded DNA, and it's a pseudo-retrovirus. Meaning, this double-stranded DNA, when it replicates, it goes through an RNA intermediate, and like you have probably heard of, say, HIVs and other retroviruses, this RNA is reverse transcribed back into the cDNA. But when it gets reverse transcribed, the two strands that are made, the negative strand is complete, whereas the positive strand is incomplete. And that's the reason why this particular viral genome is partially double-stranded. So once this virus gets inside the cell, and there are many unresolved areas in animal virology, and here also HBV shows a very strict hepatotropism, meaning that hepatitis B virus can only infect liver cells, not any other cells. And the reason for that could be like the receptor that the liver cells, the receptor that it has got can only recognize hepatitis B virus, but in other cells the right receptors are not there for necessary attachment of the virion onto the cell surface. So once it gets attached to the cell surface, it releases the nucleic acid, okay, the nucleocapsid onto inside the cytoplasm. And then this is again a question mark. There are competing ideas, hypotheses whether the completion of double-stranded DNA genome happens in the cytoplasm or it happens in the nucleus. So the opinion is divided. But to make a long story short, in the nucleus we have a double-stranded circular DNA genome like you see in a plasmid. Interestingly, the size of this genome is only 3,200 base pairs, almost like the regular cloning plasmids that we use, like say almost the size of say blue script, probably you have heard of the name, the cloning vector. So it's almost the size of this blue script vector. But the problem with this particular double-stranded DNA is, if you look at the world population, probably 1/4 of world population is either been infected or a carrier of hepatitis B virus. In Southeast Asia, in almost entire China, is the carrier, all Chinese population is carrier of hepatitis B virus. And as because it only transmits through the body fluids, so every mother to child, every childbirth, so the next generation will be infected with hepatitis B virus. So with vaccination, this number is coming down, but we do not actually know the mechanism through which hepatitis B virus can actually attain this level of success with such a small genome. And the work that we are doing here is basically trying to address the issue of how we can somehow stop the replication of hepatitis B virus. So that is our goal. Okay? But we are very far away from there, but that is the reason why we do this particular work. Okay, so once it's inside the cell, it makes RNAs. And this is, and I will show you in detail in the next slide. So they actually make a RNA which is longer than its genome size. The reason it is longer than its genome size is probably if you have, if you know about say eukaryotic transcription, so there are certain polyadenylation that takes place when the eukaryotic RNA, it's at the end of those RNAs, and hepatitis B virus utilizes the host RNA replication mechanism. So once this RNA is synthesized over here, it gets out of the nucleus to the cytoplasm. So that's exactly where we will be working, how this RNA gets out. And when this RNA gets translated, and they will make the viral proteins, and eventually in the cytoplasm it will make a nucleocapsid, and that nucleocapsid will be then covered by the surface antigens, virally encoded surface antigens, and these surface antigens will bud out from the hepatocytes. So once this mechanism is established, this hepatocyte becomes a living factory of new hepatitis B virus particles. Okay. So the problem is, once hepatitis B virus gains entry inside into the cell, you cannot actually take it out from here. So no matter whatever drug that you do, that you give, it's only going to act inside the cell, and basically you have to go for an early level therapy that you go for say cancerous cells. As because it employs the host machinery, the treatment is giving you the nucleotide analogs. So once those analogs get inside this, this DNA structure will be altered, and the hepatitis B virus will not be able to replicate. But basically, you have to go for the first line of anti-cancer therapy, and that's the reason why the treatment of hepatitis B virus is not always very kind of easy on the individuals. Okay, so this is how the virus would look like under electron microscope, and this is the whole thing that I was talking about, that it has got a full length negative stranded DNA, but the positive strand is incomplete. So once it gets inside the cell, inside the nucleus or cytoplasm, So this will need to be completed. And again, it is not essentially, nucleotide to nucleotide binding, because it has got some RNA component bound to the positive strand, and there is the polymerase component. Polymerase, the reverse transcriptase, is actually bound to the end of the negative strand. And if you look over here, so these are the coding regions for different viral proteins. This blue one is the polymerase. This red one is the X protein, which is a pleiotropic regulator of cell signaling. So it acts at many different levels. And there is a core protein which is responsible for forming the nucleocapsid structure. And then there is the surface antigens, which comes in three varieties. One is called the long surface antigen. So that starts from here. And then you have another translation start site that makes this medium-sized surface antigen, on which upon proteolytic digestion, the small surface antigen comes out. And all these combination of surface antigens are responsible for making the coat of the hepatitis B virus. And again, there are many variations. Only the ones which forms this kind of casings are called of a 42 nanometer size. We call it the DEN particles. And these DEN particles not necessarily will always contain the nucleic acid inside. Probably only 10 to 20% of these DEN particles will have nucleic acid in them. So the idea is, if you have somehow generated antibody against the hepatitis B virus, these antibodies will be absorbed by these empty shells, so that the shells which contain the nucleic acid inside will be free to infect. So that's again another strategy of spreading the infection. And this is how the different RNAs from this encoded by this double-stranded circular genome is going to look like. As I mentioned initially, if you look at this black one and this red one, these two RNAs are actually longer than genome length. The reason they are longer than genome length is that if here is the polyadenylation signal, when transcription starts from here or here, during the first passage, the RNA polymerase II transcription does not terminate over here, and it goes all the way around to be terminated on the second pass. But for transcription beginning from here or from here, the polyadenylation takes place during the first passage itself. So the exact mechanism why, when transcription starts from this particular promoter or this promoter, does not terminate at this particular polyadenylation signal, or this polyadenylation cleavage is not taking place, is not immediately obvious to us. And this is again a gray area, but it is central for the hepatitis B virus to make these two very important RNAs for itself. This red one is the long X RNA, and this black one is called the pregenomic or precore RNA. Our research interest is basically on this black one, which is this pregenomic RNA. And because this pregenomic RNA will be used both as a template for translation of this polymerase protein, and as the name suggests, this particular this pregenomic RNA will again be used as a template for making the new viral genome. So these particular RNAs exit from the nucleus to the cytoplasm is paramount to the replication of hepatitis B virus RNA, and we are interested to know what mechanism does it employs to get out of the nucleus. So let me just brief you a little bit about, if you are not familiar, I think many of you would be. So there are different export pathways for RNAs to get out from the nucleus. mRNAs generally employ a subclass of molecules which are coupled to its maturation. For example, so if this is the blue one is your RNA and it contains the 5' cap, there are a bunch of different complexes which assemble on this particular RNA, which is dependent on how this particular eukaryotic mRNA gets matured. So what do I mean by this? As you all know, in case of say eukaryotes, splicing is an important part because the gene is much way longer than the mRNA itself. And the reason these mRNAs get shortened is through a process of splicing, meaning several pieces of RNA will be excised out from there and then this RNA will be joined together. Okay. So once this splicing takes place, it's not only that the RNA, some piece of RNA is being excised out from it, it has also got a very serious connotation about the transcription going. So everything that happens during synthesis of RNA, messenger RNA, every maturation step is actually co-transcriptional, not post-transcriptional. Meaning, it's not that you make the RNA, then take pieces out, then add the poly A tail, it doesn't occur that way. Unless you add the 5' cap, the RNA polymerase will not move beyond say 20 bases. So addition of cap is a signal for the RNA polymerase to go further into the elongation mode. And as it goes, the entire RNA polymerase is actually a factory, is kind of a dynamic factory that many factors come and join, leave the spot, new proteins come and like do the next step. So the RNA polymerase that starts from here and the RNA polymerase that ends up at the end of the gene are two very different molecules. They only share certain components, but all the decoration that it has on top of it is very different. Similarly, as the RNA polymerase synthesizes the RNA and all other such maturation processes are taking place on it at the same time, so does the assembly of the export complex. And this TREX complex is an important component that needs to assemble on the RNA itself, guided by the cues of splicing being taking place on these RNAs. And then, so these are East nomenclatures, two export adapters which are called NXF1, NXT1 in metazoans and MEX67 and MTR2 in East. They should also associate with this mature mRNA. Now the question is, it was, say, if you are discussing this problem about say 2 decades back, the idea was splicing acts as a licensing mechanism so that splicing will deposit certain proteins which will license the RNA to get out of the nucleus. Now as we understand it, that export is a default process and there must be mechanisms in the cell that should restrict free passage of RNA from nucleus to the cytoplasm. So what I am meaning is that for export, there is some sort of a kind of a quality control that should ensure only the spliced RNA leaves the nucleus and is ready for translation. And the unspliced RNA must remain within the nucleus. Now, in case of say hepatitis B virus RNA that we talked about, here there are certain introns inside, we will show it. So, if the RNA, this pregenomic RNA needs to go out of the nucleus, then it should also bypass the surveillance mechanism present in the cell that inhibits the passage of the intron containing pre-messenger RNA into the cytoplasm. So that's where the story gets a little murky. And then, so when it gets into the cytoplasm, these things, I mean, the pattern of ribonucleoprotein, the proteins that are associated with this RNA will change entirely. So this will be again recycled back into the nucleus and go for the next round. Now, this was a story for say mRNA, right? But for say other RNAs, say USN RNA, large ribosomal RNAs, 5S RNAs and tRNAs, another particular protein which is called like the major family of karyoferrins. Karyo means nucleus, ferrins means which ferries it. So this is called CRM1. This particular karyoferrin, it interacts with all these other different types of RNAs that is present in the nucleus. So mRNAs distinguish themselves by being exported through this NXF1, NXT1 pathway, whereas other different types of all the RNAs, they go through by the CRM1 pathway. And obviously, as because whether you can recruit CRM1 or you can recruit NXF1, NXT1 is a large kind of policy decision for any virus or any mRNA that needs to get exported. So, retroviral RNAs, we say, hijack cellular pathways for nuclear export. They do not encode any protein that is directly involved in export. So, as because it is using the host machinery, so it needs to have some sort of kind of plan to use the host machinery to export it out. Take for example, say HIV mRNA. So this mRNA also contains introns, and these introns, this intron containing RNA needs to get out of the nucleus. What it does, it actually codes for one protein which is called REV, and this particular viral protein REV can bind to certain regions of its own RNA, which are called REV response elements, and binding of REVs to those RRDs, and along with the the helicase DDX3 can actually recruit CRM1. So if you can recruit CRM1, your job is done, because cell is ready with all its other partners to bind to CRM1, and it could get exported out of the nucleus. Whereas in case of say regular cellular mRNA, the CRM1 is not involved. So this TAP next is another previous nomenclature of this NXF1, NXT1. So this TAP NXF1 has got many names, MTR2, TAP, etc., etc. But basically they are the same proteins, and they will then bind to the nuclear pore complex. Once they get associated with this basket, then there will be some equilibration going on, and finally they will be released. Again, another retrovirus, which is called MPMP, Meloni, Pfizer, I forget the big name, whatever. So it is again a retrovirus. It contains one element which is called CTE, co-transcriptional transport element, and this co-transcriptional transport element can bind to TAP. TAP means the one that is responsible for mRNA export. So this MPMB RNA will come through the TAP pathway. Whereas the HIV RNA, both are intron containing. One comes through the your NXF1, NXT1, the CRM pathway, and this particular RNA comes through the TAP pathway. So there is no thumb rule that all the RNAs will come through one particular pathway. Different RNAs will employ different mechanisms to get out. So in HBV field, for the last 20 years, people are fighting on this. Which pathway, say 10, 15 years back, it was almost unequivocally established that the NXF1, NXT1 pathway is the one to come from one method of choice. But about 6 to 7 years back, one group showed that no, CRM1 can actually bind to these RNAs through these A-rich elements present in the poly-A tail, and binding to this poly-A tail will qualify this RNA to go through CRM1, and that can be inhibited by another cellular protein called E-level1. So HBV RNA necessarily employs the CRM1 pathway. But about 20-25 years back, it was shown that if you treat the cell with leptomycin B, which is an inhibitor of CRM1 pathway, that does not block the export of the HBB RNA from nucleus to the cytoplasm. So the jury is always out. And that's where we tried to biochemically address this issue by looking at different cellular proteins that can interact with it and what result can we get based on the biochemical and microscopic assays of which pathway our minigene RNA, which is being made from the pregenomic RNA, is going to go through. So first of all, the cellular proteins which are interacting with the HBV pregenomic RNA, surprisingly the GAPDH, a particular protein that is we chose to work on, I'll elaborate on why. That's called polypeptide tract binding protein or PTB, and the La protein, and another protein is, I mean, without any known function, so to say, that ZC3H18, a cellular protein recruited constituents of the TREX complex, as I showed, the TREX complex which needs to be recruited onto the RNA for committing itself to the NXF1 NXT pathway to a sub-element of pre. And pre, like we have seen for say CTE over here, co-transcriptional transport element, here pre is another component in a hepatitis B virus which is called a post-transcriptional regulatory element. And this is the structure of polypeptide tract binding protein or PTB. It has got 4 RRMs, RNA recognition motifs, in which RRM2 is already established to be a splicing inhibitor, but the role of other 3 RRMs are uncertain. Okay. Now, what we'll be trying to do over here is to look for different sequences in the pregenomic RNA, in our minigene constant in the pregenomic RNA, and whether they can interact with polypeptide tract binding protein, and how this interaction with polypeptide tract binding protein interferes with the nuclear export. So this is the minigene we are working with. We call it CPS pre because it contains the coding regions of these core polymerase and surface antigens, and we have added this post-transcriptional regulatory element pre over at the 3' end of this particular gene. And it has got, the splice, it can be spliced, but it is not really spliced very efficiently. And this is another challenge why the viral RNA doesn't get spliced and the pregenomic RNA always comes out as the unspliced one. So it contains these intronic regions intact, and these are their sites. And with respect to the RNA, this is the different coding regions, the core polymerase or S1, S2, S, etc. And there are 2 polymerase domains, which is polypyrimidine domain, which is called PPD1 and PPD2. This PPD1 is present within the intron, and this PPD2, this polypyrimidine domain, is present in the pre, that is in the 3' exon. And we have also checked with another variety of the same RNA where this intron is spliced out to look for any specific role the presence of intron or the event of splicing has on RNA export. Okay. So first of all, what we did some biochemical assay by measuring the presence of this particular minigene RNA either in the cytoplasm or in the nucleus and did their ratio. So CPS pre is our basal construct, and we gradually made different deletions in these polypyrimidine domains, and this polypyrimidine domain 1 is, as I said, present in the intron, and polypyrimidine domain 2 is present in the pre in the exon. And one thing I just want you to remember right now, but we'll come to that later. Within this pre, which was supposed to be the export promoter, or which is upregulate export, there's a region in pre which is called beta, is supposedly one that is also specifically involved in this upregulation. And every time we tinkered with this polypyrimidine 1 sequence or polypyrimidine 2 sequence, it increased export. So essentially what that means is that presence of this polymeridian domain was responsible for inhibiting export. Okay, and you want the, because it's a intron containing RNA, this polymeridian domains was responsible for keeping the RNA in the nucleus, it was preventing export. And if you look at the splicing of different such constructs, you do not see much of a difference when the polymeridian domains are deleted. Meaning these polymeridian domains are not necessarily involved in regulation of splicing. The reason we are saying this, because as I said, if polymeridian domains are recognized by PTB, and PTB generally inhibits splicing, so if you delete these polymeridian domains, your splicing should go up. The fact that it did not, meaning that the interaction with PTB or any splicing repression that might be happening on the cell is not going through these polymeridian domains. However, it's when you take these regions out, its splicing increases. And even like, when you replace these polymeridian sequences with purines, the effect is not really that great. So meaning there's a specific way these polymeridians can interfere with export. And then we looked as single molecule RNA fish. Let me just explain a technique a little bit, so that will explain why this is important. So essentially when you do fish, any RNA fish for that matter, you are not actually looking at the single molecule of RNA, you are adding one large probe, and that large probe should light up. So here, if you are looking at, if you want to look at a single molecule of RNA, and the number of that particular RNA is going to be very little inside the cell. So what you do, you make a series of oligonucleotides tiled up. So they bind one right next to the other like that. So here you are using about say 35 oligonucleotides, all bound to a single fluorophore. So here it is say TRITC. Okay. So every dot that you see represents one RNA molecule. And we are looking at how single RNA molecules, how they behave inside the nucleus and how they are going out. Okay. And what you see, in case of the CPS and the percentage of export that we calculated, so we counted the number of such dots present in the cytoplasm and in the nucleus, and then made the ratio to calculate the percentage of export. So in case of the CPS, our mean value of percentage export about say 18%. And when we add pre, which is supposedly an upregulator of export, this number goes up to say 24%, 27%. But look at these numbers. When you delete the intronic polypyrimidine domain or the extrinsic polypyrimidine 2 domain, and when you delete both, the number actually jumps about say threefold. So what we have seen biochemically, we are now seeing the visual evidence that it kind of validates our previous biochemical finding, that it does indeed increase in the amount of export of these RNAs. And this is again a cross check. So the fish probes that we used here are actually all in the introns, meaning this red signal are actually counting only the intron containing RNA that came out from the nucleus to the cytoplasm. Okay. And now here what we have done, we have used green fluorophore leveled and FITC leveled probes which were designed against the exons. So if these intron containing RNAs and exon containing RNAs, they are the same, so they should be, they should merge on each other. Okay. So this gives us proof that we are not looking at the excised introns from the RNA, but we are actually looking at the full length RNAs but containing the introns inside it. So these are with transiently transfected cells, and these data shows here with stably expressed cells, where the result is even more dramatic. You see about say 5-fold increase when both the polypyridine domains are deleted, where it was about say 3-fold over here, say 18 to 57 and 10 to 46. So when you are stably expressing these lines, you see a more dramatic result, and almost like, the DAPI stain of nucleus is now more clearly visible as blue, but in all other cases, as because the blue was merged with red, so it's kind of hazy, but here you see like, and we have better pictures for those later on. So the export increases. Now, why do we think PTB is important? And we then previously showed biochemically that, that PTB could associate with PPD1 and PPD2 domains. So unless you can show the ability of polypyridine tract binding protein to these polypyridine domains, there is no point. So essentially what we have done, we first did some RNA immunoprecipitation assay. So from the cell itself, we used the antibody against PTB, pulled the whole complex down after cross-linking, and tried to look for the presence of these RNAs. So what we find here, that if you delete the intronic polypyrimidine domain 1, the binding of the PTB with the RNA is destroyed. So this is the primary focus or primary point through which the PTB makes contact, and if you can destroy that, further contacts will not be possible. But if you delete other polypyridine domains, you do not really see that much effect, meaning that if PPD1 is present, it's kind of a nucleation of PTB, or making a toehold over there and then spreading to the other regions. And this shows the purity of the PTB that we overexpressed, which was histidine labeled. And then we did some UV cross-linking assay in presence of say different competitors. And obviously it shows like, the PPD1 to which when our protein PTB binds, it shows a very strong interaction, and that is evidence through this competitor PPD1. And this PPD1 pyrimidines, when they are replaced by purines, it did not compete much, meaning the binding was pyrimidine specific, and PTB could bind to the polypyridine domain over here. And then in D, we see the ability of PTB to bind to both PPD1 and PPD2. Okay. So these polypyrimidine domains are legitimate polypyrimidine domains to which PTB can bind. And then what happens if we now overexpress PTB in the system? We are increasing the amount of PTB in the system, so the export should be compromised. That's exactly what we see. So PTB 4 MST means where, okay, so let me just explain the assay a little bit. So what is happening over here, we have now, so how do you artificially dock one protein on the RNA? So there is one particular system which is called MST coat protein binding site. So if your RNA has a MST coat protein domain, and you can express the MST coat protein domain on your protein, so binding between that RNA sequence and MST coat protein domain will tether the protein onto the RNA. And so here we have made these PTB constructs. It contains all 4 domains of PTB. Here domain 4 is missing, and here only 2 is present, and this is the control. So you see, like, this is all PTB variants with different RRM domains present in there. In all these cases, you find that when we express PTB4MS2, that export is almost lost. Okay, but as you go on, like, using other PTB domains, every time you do away with the RRM4 that absent from an expressed protein, this inhibition of export is lost, meaning RRM4, which is not reported so far, that RRM4 is probably responsible for suppression of export that we see over here. And this one shows like deletion of PPT1 and 2 domains is consequential to splicing, because every time you overexpress PTB, splicing diminishes. And then we compared the same scenario when a single molecule RNA fusion was done under PTB knockdown scenario with SIPTB or overexpression. So if you just look at this last panel where both PPT1 and PPT2 is absent, okay, and what you are doing here, and you are taking the PTB out as well. So you can see the amount of exported, which was 60% of the RNA is getting exported out. Whereas over here, when you, the 18% that we have seen for the CPS pre when PTB was present inside the cell, if you take the PTB out, that jumps to say 51%. Meaning that PTB is indeed responsible in the pathway of regulating or suppressing the export of the pregenomic RNA in the nucleus to the cytoplasm. And over here, where both domains PPT1 and PPT2 was replaced with MS2, and you add MS2 PTB4 or MS2 PTB2L, you can see like the presence of PTB4 actually suppresses it, but if you use the 2L where the RRM4 is absent, this particular suppression is lost. So that meaning that RRM4 in PTB is involved in regulation of export, whereas RRM2 is involved in regulation of splicing. And then so the next question that we were asking, so between two export pathways, the NXF, NXT1, which is involved with the pre-mRNA export pathway, and the CRM1, which is used for export of say, other RNAs, which one does actually our minigene pre-mRNA choose? So we have done the similar experiment, but it is an RNA fish coupled with immunofluorescence. Meaning we are looking at the RNA through immunofluorescence, and we are adding antibodies against different export factors, and the secondary antibody contains the FITC. So the green fluorescence is coming from the protein part, and the red fluorescence is coming from the RNA part. And we see that CRM1, if you do for the CRM1, there is not really any specificity of co-localization of the RNA with that of the export factor. But when you go with say NXF1, you will find like most of the NXF1 signals are now merged with where the RNA signals are. So that shows us like microscopically that this particular RNAs, this CPS pre, is in a position to interact specifically with NXF1. The same is true with NXT1, and we also checked with the other members of the NXT1 pathway, that is THOC2 and THOC5. We see similar results in all the cases. So that shows like microscopically we can actually put only the members of this NXF1 NXT1 pathway close to the RNA, not the CRM1. And these are for say all other members of say this NXF1 NXT1 pathway. I am not going into the details of it. Now again the biochemical angle to it. So when you do the immunoprecipitation, RNA immunoprecipitation with CRM1, no RNA could be actually found bound to it. Whereas when you do it say NXF1 or NXT1 or say all other members of the pathway, you can find them attached to the export factor, export adapters. So that again shows both biochemically and microbiologically that this particular RNA actually chooses the NXF1 NXT pathway over the CRM1 pathway. And as I said, like if you remove the beta region, which is specifically implicated in pre to upregulate transcription. So if you delete the beta region, so over here you can see in all the cases deletion of beta completely inhibits the export of this pregenomic RNA. So based on that we can actually conclude a model. So here in this case, PTB initially binds to this beta and these PPDs, which are very close by the way, and binding of PTB to this PPD1, which is the intronic one, is central to the recruitment of this particular PTBs on these other regions, and the assembly of the TREX complex over here by all these other members that we have shown to be associated with it can actually dislodge this particular PTB from this PPD2 region, allowing this beta region to sponsor the binding of the NXF1, NXT1. That is our model. And this particular RNA, when that happens, will be exported out. But if on those particular RNAs where this TREX complex is not able to displace PTB from the PPD2, these RNAs will remain inside the nucleus. So essentially, if you can actually inhibit this process where PTB mediated inhibition of keeping the pregenomic RNA inside the nucleus, you can actually stop the passage of the pregenomic RNA of HBV virus to get into the cytoplasm. And once you do that, you can actually inhibit HBV replication. Because in case of say carriers, you have not been able to stop the replication. You are only inhibiting when they are being replicating, and as a result, you are giving anti-cancer drugs. So here, you don't really have to go for these nucleotide analogs. You can pinpoint one particular step in the export pathway through which you can inhibit RNA export. So essentially, this is about say PTB RRM2 was not effective in inhibiting transport as much as the PTB, but RRM2 was responsible for inhibiting splicing. So we are finding another new role for other RRMs, that is specifically RRM4 in PTB, and this particular binding. So let me stress on one point over here. So the question that was initially asking, so what is the pathway through which the intron containing RNAs are inhibited at the nuclear periphery? So there has to be some sort of a quality control that will not allow the presence of introns in the RNAs that gets into the cytoplasm. And this MLP1, PML39 and its homologs in metazoans are actually such gatekeepers which look for the presence of introns, and if you have it, you are not allowed to pass on. So that is done. So this assembly of NX7 and NXT1 by PTB probably somehow licenses these intron containing RNAs for export. And here this PTB mediated export inhibition must somehow circumvent this problem. And from a therapeutic point of view, this PTB mediated inhibition of nuclear export of intron containing RNA can also provide a handle to control HBV replication. And these are the two research scholars who have actually worked on this project. This work is supported by the grant from DBT and of course the Department of Microbiology, University of Calcutta, for providing all support. Thank you very much for your attention.
Thank you, Professor Ghosh. From your study, it appears that we can hope for the development of some inhibiting mechanisms for the replications to inhibit the replications of the virus. Okay, so you have shown that different types of models. Like PTB, then PPD1, PPD2, and NRX and NXT models are also involved there. So that is very nice presentations. I appreciate that.
But we have still a long way to go.
No, yeah, we can, because hepatitis. For the hepatitis, I don't think that I should give the scope to the audience and other dignitaries. If they have any questions, they can answer. Then after
Sure.
that, I'll.
Sure, sure.
Thank you, sir, for your high standard lecture on your innovative studies. I have got some simple questions. First thing, TREX binds to the maturing mRNA, but ultimately it is getting detached from the mRNA. What is the exact time that TREX will get detached, and which phenomenon or which matter induces this dislodgement of the TREX from the RNA? One thing. And second thing, is there any therapeutic drug, antiviral drug or any other drug that can actually inhibit the entire, any step of, can stop any step in nuclear transport?
Excellent question. So that's exactly only for say last 3 years back, and on the verge of say so many different molecules being tried on this field. Only for the last 2 or 3 years, people have started exploring this export pathway as a target. It generally has been replication or transcription, because it's easy to make different types of variant nucleotides. You can have a battery of different nucleotides and check whether that works or not. But the moment you go for a very specific point in a pathway, the chemistry becomes a lot more difficult and there's a lot of guesswork. So only for say last 2 or 3 years, this particular checkpoint, coming to your first part of your question, where exactly TREX gets detached. So the idea is, so over here, when this mRNA and this complex is presented to this nuclear pore, so these are called export adapters for a reason that these adapters will dock the cargo, the RNA protein complex, onto this particular gate. So once it gets docked over here, then the structural changes will take place, and that's the reason why it's an equilibration through nuclear pore. Conformational change will, as I said, the RNA biology is nothing about sequence. It's all about structure. You change the structure, one group of proteins will bind, and you change another, go to another structure, another group of proteins will bind. So this constant structural alteration of the protein mediated by these RNA binding proteins will either make it bind to something, or the affinity will get reversed and somebody else will take over. So that's where the kind of this decoration on the RNA changes at the nuclear pore complex. And this East MLP1 that I was talking over here is present inside it, in this nuclear basket, pore basket. So in this nuclear pore basket, if the intron is present, and we say as the intron is present, we are not really sure whether the intron is being recognized or certain proteins which binds only to introns. And this is again very difficult to differentiate between the cause and the effect. If this protein is there because the intron is there, or because the intron actually created.
TREX comes out at a site where intron ends, or what?
No, this RNA contains intron. So basically, if the splicing happens, that means intron should be out, and then the TREX complex should recognize this event of splicing and will be recruited. But somehow you have certain protein which can actually, like we have seen for say MPMB or say RAVE, if certain protein can recognize an alternative structure and recognize the TREX complex, then TREX is fooled as if to say that splicing has happened, but the splicing has not happened. Because in the next step is only TREX is going to be recognized, not the intron. মানে আপনি যদি কোনো ক্রোমেটাকে ঘুরিয়ে ট্রেক্স রিকগনাইজ করে দিতে পারেন তাহলে কিন্তু চলে যাবে। So this kind of like fooling the system of recognition of different proteins. So our idea was PTB actually does this job. PTB does not allow the TREX complex to bind to it when it is bound to PPD1. So that's how PTB inhibits export. So if you take the single 6 nucleotides out, then PTB cannot bind because that is a nucleation site.
It cannot be used as a drug, PTB?
PTB is an essential protein. I mean, for most of our RNAs, cellular RNAs, splicing is regulated by PTB. Splicing is regulated by PTB. Only hope is that RRM4 is not responsible for any other cellular effect that we know of. But here RRM4 is important. So if we can design something against the RRM4, then maybe something will happen, but these RRM2 related events will not be touched, because we do not know any cellular process which goes through RRM4. So if you can actually make use of this RRM4 domain, then you can actually look for PDB related efforts. But one hitch is there. Every time we overexpressed whole PTB, the entire cellular export pathway got suppressed. So RRM4 probably has role anywhere that our PTB binds. RRM2 is involved with recruitment of events related to splicing. RRM4 probably binds to the other export factors through some indirect interactions, and the players are unknown in that area.
Thank you.
Sure.
Excellent work, Subhadra. I have two basic questions. So first question, this virus, whether it's only restricted to the hepatocytes, or it can also go to the other liver cells?
No. There are two reasons for this. One is the specificity for the receptor, and secondly, these promoters require certain hepatic proteins which are not expressed elsewhere. Because it's totally host polymerase that does the transcription. So the assembly of pre-initiation complex in this covalently closed circular DNA of hepatitis B virus can only happen with hepatocyte extracts. No other cellular extract will be able to transcribe.
And the second thing also, whether these receptors that express into the hepatocytes, those are depending on the hepatocytes physiology, or do they express constitutively?
Probably constitutively. Probably constitutively, because from the there is no kind of age barrier or defined developmental barrier at which HBV cannot infect. So even from the womb itself, you get infected, and till the very end, you can always get infected. So I would say, there is no such developmental barrier. Something very constitutive element is being recognized, which is unique. So the moment hepatocytes are differentiated, this receptor is there, and that's happening.
Thank you.
So I think we should conclude this session. And thank you very much, Professor Ghosh, for your nice talk.
It's my pleasure.
Our profound thanks to all the dignitaries present here. Your presence and wisdom have made this session truly enriching experience. So now we have a lunch break, and next we will return back to our PG and UG poster presentation at second and third floor of Pure and Applied Health Science building. Thank you.
Hello. अच्छे साजो अभी सारे पहलू सुनील बेस काजर नयाने सलाज बयाने कुसुमे सजानो के सखी हम मोहन अभी सारे जाऊं बोलो हम एतक सुख कहाँ पाऊं
কম্পিত তনু দল গুঞ্জিত বাদল পুঞ্জিত শ্যামল মালা কম্পিত তনু দল গুঞ্জিত বাদল পুঞ্জিত শ্যামল মালা নল কিশোর মম কোমলন নিরসম নল কিশোর মম কোমলন নিরসম ক্ষণে গোরা চাঁদ ক্ষণে কালা ক্ষণে গোরা চাঁদ ক্ষণে কালা কম্পিত তনু দল গুঞ্জিত বাদল পুঞ্জিত শ্যামল মালা
ওয়াও থ্যাংক ইউ সো মাচ আমাদের এত সুন্দর একটা পারফরম্যান্স গিফট করার জন্য নেক্সট পারফরম্যান্স এমন একটা সঙ্গের ওপর যেটা শুনলে মনে হয় আমি ক্লাসিক্যাল শিখিনি কেন সো আর বেশি কথা নয় প্রেজেন্টিং প্রীতি দিন্ডা ইউজি 5th সেম ডিপার্টমেন্ট অফ নিউট্রিশন
रघुकुल रीत सदा चली आई प्राण जाए पर वचन न जाए
থ্যাংক ইউ প্রীতি। ওকে ওকে, ডান্স ফ্লোর এবার একটু বিশ্রাম নিক, কারণ এবার ভয়েস টেস্ট হবে। ইটস টাইম ফর লাইভ মিউজিক ম্যাজিক, অরুণিমা অ্যান্ড চন্দ্রিমা অ্যান্ড অর্কনীল, যার গান শুনলে মনে হবে স্পটিফাই কিসের দরকার। সো আর দেরি কিসের, ক্লোজ ইউর আইজ অ্যান্ড ফিলস দ্য ভাইব। শুভেচ্ছা একটু এখানে আসবে। যারা এখনো নিজেদের স্ন্যাক্স নাওনি তারা ক্যান্টিনে চলে যাও, নিজেদের স্ন্যাক্সটা নিয়ে নাও।
Hello, 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 hello. Hello. Hello.
Hello. উড়বো বলে
কে জানে এই আমায় দেখে হঠাৎ এমন চমকে ওঠার মানে সে তো সবাই জানে অঙ্ক কঠিন মিলবে না একটাও সবাই জানে অঙ্ক কঠিন মিলবে না একটাও আমার তোমায় দেখে আশ মেটে না কোথায় যাবে যাও আমায় অন্ধ করে দাও এবার অন্ধ করে দাও দৃষ্টি কেড়ে নাও গো আমায় অন্ধ করে দাও আমায় অন্ধ করে দাও আমায় অন্ধ করে দাও তুমি তো সব দাও গো আমায় অন্ধ করে দাও মন ভেঙে দাও পাহাড় থেকে ঠেলে আমি খুন খারাপি চাই না এমন মন খারাপে পেলে কেমন বেয়াক্কেলের শুনলে কথা জ্বলবে তোমার গাও বেয়াক্কেলের বড্ড জ্বালায় আর একটা গান গাও আমি জ্বলবো তবু ছাই হব না সেই বাঁশি বাজাও আমায় পাগল করে দাও এবার পাগল করে দাও উড়ন্ত সব নেশার মতো পাগল করে দাও আমায় অন্ধ করে দাও আমায় অন্ধ করে দাও তুমি তো সব দাও গো আমায় অন্ধ করে দাও লক্ষ বছর ঘুম আসেনি জল জোটেনি ঠোঁটে তুমি আমার পেটে মারলে লাথি রাগ হবে না মোটে আমি ফুল ফোটাবো না হুল ফোটাবো কোনটা তুমি চাও ফুল ফোটাবো না হুল ফোটাবো কোনটা তুমি চাও খেলাতে বারণ চোখের পলক ফেলা চোখের জল আটকাও আমায় ধ্বংস করে দাও এবার ধ্বংস করে দাও উড়ন্ত সব নেশা ধ্বংস করে দাও আমায় পাগল করে দাও আমায় পাগল করে দাও উড়ন্ত সব নেশার মতো পাগল করে দাও আমায় অন্ধ করে দাও আমায় অন্ধ করে দাও তুমি তো সব দাও গো আমায় অন্ধ করে দাও
হ্যালো, সত্যি সত্যি কি সবাই পাগল হয়ে গেলে? জানি অঙ্ক বড্ড কঠিন, তা বলে এত চুপচাপ? আচ্ছা, বুঝেছি। এবার আরও একটা ডান্স পারফরমেন্স চাই, কি তাই তো? আচ্ছা, তাহলে চলো, আর বেশি দেরি নয়, আমরা MBA ডিপার্টমেন্ট থেকে ডেকে নিচ্ছি শুভশ্রী অধিকারীকে। সরি, শুভেচ্ছা অধিকারীকে।
প্রিয় চন্দন টিকা তার মাথে আজ পরে নাও পরে নাও পরে নাও আজ নাই গুনগুন গুঞ্জন প্রেমে চাঁদ ফুল জোছনার গানার নয় ওগো প্রিয় মন খোলো বাহু ডোর পৃথিবী তোমারে যে চায় কার ঘরে প্রদীপ জ্বলেনি কার বাঁচার অন্ন মেলেনি কার নেই আশ্রয় বরসায় দিন কাটে ভাগের ভরসায় কার ঘরে প্রদীপ জ্বলেনি কার বাঁচার অন্ন মেলেনি কার নেই আশ্রয় বরসায় দিন কাটে ভাগের ভরসায় তুমি হও একজন তাদেরই কাঁধে আজ তার ভার তুলে নাও তুলে নাও তুলে নাও আজ নয় গুনগুন গুঞ্জন প্রেমে চাঁদ ফুল জোছনার গানার নয় ওগো প্রিয় মন খোলো বাহু ডোর পৃথিবী তোমারে যে চায় আজ নয় গুনগুন গুঞ্জন প্রেমে চাঁদ ফুল জোছনার গানার নয় ওগো প্রিয় মন খোলো বাহু ডোর পৃথিবী তোমারে যে চায় পৃথিবী তোমারে যে চায় পৃথিবী তোমারে যে চায়
কি স্নিগ্ধ। থ্যাংক ইউ, শুভেচ্ছা চারিদিকে এত স্নিগ্ধতা ছড়ানোর জন্য ধন্যবাদ। এবার আমরা দেখতে চলেছি একটি দারুণ সুন্দর এবং এনার্জেটিক মিক্সড ডান্স পারফরমেন্স। ডান্স মানেই রিদিম এক্সপ্রেশন আর পিওর এন্টারটেনমেন্ট। আর আজকের এই পারফরমেন্স আপনাদের সামনে তুলে ধরবে একটি সুন্দর গল্প মিউজিক আর মুভমেন্টের মাধ্যমে। তাহলে আর দেরি কেন, লেটস ওয়েলকাম দিশা, সুপর্ণা, শান্তনা, অনিন্দিতা, সুরশ্রী, তৃষা আর সহেলি।
खिला है चाँद।
a girls' party. What a swag! এবার আসছে একটু বলিউডি ফিল নিয়ে সুকন্যা পান from PG 4th SEM Food Science and Nutrition performing a solo dance on Nano Alene. Let's clap for her.
चैन रैन नैन अपने साथ ले गया नैनो वाले ने छेड़ा मन का प्याला छलकाई मधुशाला मेरा चैन रैन नैन अपने साथ ले गया पग पग डोलो रे हो पग पग डोलो रे डगमग सी मैं चलती हूँ जगमग लो सी जलती तेरे नैनों की कैसी मदिरा थर थर कांपू रे तेरे तीर से झिपती चंदन पे नाग सी लिपटी मैं बेहोश तू नशा ऐसी मोह की दशा मेरा चैन रैन नैन अपने साथ ले गया नैनो वाले ने छेड़ा मन का प्याला छलकाई मधुशाला मेरा चैन रैन नैन अपने साथ ले गया
Thank you Sukarna for your lovely performance. আচ্ছা ইন্ডিয়া মানে কি শুধু কালচার একটাই কালচার না একদমই না ইন্ডিয়া মানে কত্থক গারবা ভাংরা লাভানি সব একসাথে ইটস লাইক ইন্ডিয়ান থালি সব স্বাদ এক প্লেটে এবার একটু সাইলেন্স দরকার আরে না না এক্সাম সাইলেন্স না হার্ট টাচিং সাইলেন্স বিকজ এবার একটা ইন্ডিয়ান থালি পারফরমেন্স হবে
ভারত আমাদের দেশ এখানে প্রতিটি অঞ্চলের সংস্কৃতি আলাদা ও বৈচিত্র্যময় ভারত আমাদের দেশ এখানে প্রতিটি অঞ্চলের সংস্কৃতি আলাদা ও বৈচিত্র্যময়। আজকের এই নাচের মাধ্যমে আমরা ভারতের কয়েকটি রাজ্যের সংস্কৃতি একটি মঞ্চে তুলে ধরার প্রয়াস করছি। শিল্প, আবেগ আর সংস্কৃতির প্রাণ আমাদের বাংলা, পশ্চিমবঙ্গ।
বাংলায় আমার সর্ষে ইলিশ, চিংড়ি কচিলাও, বাংলা পার্শে মাছকে ধুয়ে জিরের বাটায় দাও।
দক্ষিণ ভারতের শাস্ত্রীয় সৌন্দর্য, তামিলনাড়ু থেকে ভারতনাট্যম।
ধিম তাদারে দানি ধিম তাদারে দানি ধিম তানা দেরে না ধিম তাদারে দানি ধিম তাদারে দানি ধিম তানা দেরে না ও দানি দের দের তানি তানা দের না ও দানি দের দের তানি তানা দের না ধিম তাদারে দানি ধিম তাদারে দানি
ছন্দ ও ঘূর্ণি নৃত্য উত্তরপ্রদেশের কত্থক।
ওরে পিয়া রে গা গা মা মা পা 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 মা গা রে গা গা রে রে গা গা রে মা মা গা পা পা মা গা গা মা নি রে গা গা মা মা গা মা দা রে দা রে দা রে नजरें बोले दुनिया तोले दिल की ज़बान है दिल की ज़बान প্রিয়া
ভক্তি ও উৎসবের ছন্দ গুজরাটের গরবা।
घूमे गली चली कहाँ चोरी चोरी हवा से है बंधी डोरी रे घूमे गली चली कहाँ चोरी चोरी हवा से है बंधी डोरी रे शर्म छोड़ी छोड़ी थोड़ी नजरों की है बोले चोरी शर्मी करनी रे घूमे गली चली कहाँ चोरी चोरी हवा से है बंधी डोरी रे
তালে তালে প্রাণবন্ত ভাব মহারাষ্ট্রের লাভানি।
लटपट लटपट कमर दामिनी अधर रागिनी हो लटपट लटपट कमर दामिनी अधर रागिनी मेरे दर आई कैसी सजी धजी देखो मेरे पिया की सावरी जिया से बावरी मेरे अंगना में देखो आज खिला है चांद ये बाजे धुन झम झम झमाते झम नाचे मन झम झम झमाते झम ये बाजे धुन झम झम झमाते झम नाचे मन झम झम झमाते झम पिंगा गपोरी पिंगा गपोरी पिंगा गपोरी पिंगा पिंगा गपोरी पिंगा गपोरी पिंगा गपोरी पिंगा।
রাজস্থানের রঙিন লোকনৃত্য ঘুমার। উচ্ছ্বাস ও শক্তির নাচ পাঞ্জাবের ভাংড়া। উৎসবের আনন্দে ভরা অসমের বিহু।
কলের কল ফুল তোকে দেখি মোর গন্ডগোল বাবাই বলে বহু বানাবো লাগাই দিল হুলস্থুল কনের কল ফুল তোকে দেখি মোর গন্ধ ফুল বাবাই বলে বহু বানাবো লাগাই দিল হুলস্থুল মই দেখি তোকে তই দেখি যে মুখে মুখে দেখা পালে তোর মন ডোলে না ডোলে ও ধনী মোর সজনী তোকে না দেখিলে মোর জিয়া না ধরে কনের কল ফুল তোকে দেখি মোর গন্ধ ফুল বাবাই বলে বহু বানাবো লাগাই দিল হুলস্থুল
বৈচিত্র্যের মধ্যে ঐক্য, এই বার্তাই বহন করে আমাদের ভারতীয় সংস্কৃতি।
আরো জোরে
বন্দে
সত্যি আমরা কতটা লাকি তাই না? আমাদের দেশে কত ধরনের কালচার, সে সব তো ঠিক আছে গাইস, বাট তোমাদের মধ্যে আমি কোনো এনার্জি দেখতে পাচ্ছি না। আর একটু জোর, আর একটু এ হই না বাত। আচ্ছা ঠিক আছে, তো চলো আমরা এবার একটু ফোকের দিকে যাই।
ফোক ডান্স মানেই মাটির গন্ধ আর এমন এনার্জি যেটা আমাদের জিমের কোনো মেম্বারশিপ ছাড়াই ফিট রাখে। সো প্রেজেন্টিং অয়ন দে, দেশি বিটস, দেশি ভাইবস, ক্ল্যাপ লাইক ঢোল বিটস।
বহুদিন তোর দেখা নাই মনের দরিয়ায়, কতদিন থাকবি দূরে ঘরে ফিরে আয়। বহুদিন তোর দেখা নাই মনের দরিয়ায়, কতদিন থাকবি দূরে ঘরে ফিরে আয়। ঘরে ফিরে আয়, আজও তোরই অপেক্ষায়। হৃদয়ের পিঞ্জিরা খান খুব যে ছটফটায়, পীড়িত করিয়ায় মনে রাঙে নাই। সোহাগের গান শোনাবো প্রেমের লহমায়। দরিয়ায় আইলো তুফান, আয় কে যাবি রে, হেসে হেসে যাবি ভেসে মদিনা নগরে। দরিয়ায় আইলো তুফান, আয় কে যাবি রে, হেসে হেসে যাবি ভেসে মদিনা নগরে। ধরো হাল শক্ত হাতে, ধরো হাল শক্ত হাতে, ভয় কি নদীর সাথে। ধরো হাল শক্ত হাতে, ভয় কি নদীর সাথে। টলবে না নৌকা ভীষণ ঝড়ে রে। মন আমার কেমন কেমন করে ও বন্ধু রে, মন আমার কেমন কেমন করে ও বন্ধু রে, মন আমার কেমন কেমন করে ও বন্ধু রে, মন আমার কেমন কেমন করে।
হোয়াট এ গ্রেস! আফটার অল এটা আমাদের MCCCON 2026-এর কালচারাল প্রোগ্রাম, তো শুধু স্টুডেন্টরা করবে এটা কি মানা যায়? আমাদের কিন্তু ম্যামরাও পিছিয়ে নেই। আমাদের অ্যাডমিনিস্ট্রেটিভ অফিসের মৌমিতা ম্যাম, এখন ওনার পারফরমেন্স আমরা দেখব। সো প্লিজ ম্যাম, ওয়েলকাম। আচ্ছা প্রগতি ব্যানার্জি, তুমি যেখানে থাকো যদি রেডি হয়ে থাকো তো আমাদের সঙ্গে একটু দেখা করে যেও। প্রগতি ব্যানার্জি।
hello, 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 hello. Hello, hello, hello.
হোয়াট এ সারপ্রাইজ ম্যাম, হোয়াট এ সারপ্রাইজ। ভেরি ভেরি থ্যাংক ইউ সো মাচ আমাদের এত সুন্দর একটা সন্ধ্যা উপহার দেওয়ার জন্য। আচ্ছা, জেনজি জেনারেশনকে সবাই বলে আমরা নাকি শুধু বিটসই শুনি। আচ্ছা, আমরা কি ভক্তি সঙ্গীত শুনি না? তোমরা বলো। এই তো কদিন আগে অরিজিৎ সিংয়ের নতুন যে গোরা চাঁদটা বেরিয়েছে, কতজন তো তোমরা রিলস বানিয়ে ফেললে, তাই না বলো? আচ্ছা চলো, তাহলে আমরা আজকে প্রুভ করছি যে আমরাও কিন্তু পারি এ টু জেড। ওই যে বলে না, জুতো সেলাই থেকে চণ্ডীপাঠ, আমরা কিন্তু সব দিকেই পারফেক্ট, তাই তো জেনজি জেনারেশন? ইয়েস। তো স্টেজে এখন নন্দিতা কর, প্লিজ ওয়েলকাম নন্দিতা।
কম্পিত তনু দল গুঞ্জিত বাদল পুঞ্জিত শ্যামল মালা, কম্পিত তনু দল গুঞ্জিত বাদল পুঞ্জিত শ্যামল মালা, নল কিশোর মম কোমল নীর সম, নল কিশোর মম কোমল নীর সম, ক্ষণে গোরা চাঁদ ক্ষণে কালা, ক্ষণে গোরা চাঁদ ক্ষণে কালা, কম্পিত তনু দল গুঞ্জিত বাদল পুঞ্জিত শ্যামল মালা। জনমিয়া দেখি নাহি হেন রূপ, জনমিয়া দেখি নাহি হেন রূপ, চাঁদের মুকুর যেন চাঁদের স্বরূপ, পরশিয়া মনোহর চিত্তজনু থর থর, মধুকুপি হেরি যথা অবশ মধু, পরশিয়া মনোহর চিত্তজনু থর থর, মধুকুপি হেরি যথা অবশ মধু, চুয়া চন্দনহার ফুল সজ্জা বাহার, চুয়া চন্দনহার ফুল সজ্জা বাহার, প্রিয় বিনে কণ্টক জালা, নল কিশোর মম কোমল নীর সম, নল কিশোর মম কোমল নীর সম, ক্ষণে গোরা চাঁদ ক্ষণে কালা, ক্ষণে গোরা চাঁদ ক্ষণে কালা, বিগলিত চিকুর মৃদু হাসে মধুর, ঝঙ্কৃত নূপুর দেহে মন, লহু লহু চুমিত বদন বিকশিত, কিঙ্কিত কিঙ্কিনি কানুরি ওর, লহু লহু চুমিত বদন বিকশিত, কিঙ্কিত কিঙ্কিনি কানুরি ওর, কম্পিত তনু দল গুঞ্জিত বাদল গুঞ্জিত শ্যামল মালা, নল কিশোর মম কোমল নীর সম, নল কিশোর মম কোমল নীর সম, ক্ষণে গোরা চাঁদ ক্ষণে কালা, ক্ষণে গোরা চাঁদ ক্ষণে কালা, কম্পিত তনু দল গুঞ্জিত বাদল গুঞ্জিত শ্যামল মালা, ক্ষণে গোরা চাঁদ ক্ষণে কালা, ক্ষণে গোরা চাঁদ ক্ষণে কালা, ক্ষণে গোরা চাঁদ ক্ষণে কালা।
থ্যাংক ইউ নন্দিতা। আচ্ছা চলো এবার একটু বলিউডের দিকে পাড়ি দেওয়া যাক। লাইটস, এক্সপ্রেশন অ্যান্ড গ্রেসের লেভেল ফুল করতে আসছে আমাদের দিওয়ানি মস্তানি দেবশ্রী দাস অধিকারী। প্লিজ ক্ল্যাপ এভরিওয়ান, না হলে কিন্তু আমাদের মস্তানি রেগে যাবে। আরেকটু জোরে, এ হুই না বাত।
दीवानी हाँ दीवानी दीवानी हो गई। मशहूर मेरे इश्क की कहानी हो गई, जो जग ने न मानी तो मैंने भी ठानी, कहा थी मैं देखो कहाँ चली आई। कहते हैं ये दीवानी मस्तानी हो गई। मशहूर मेरे इश्क की कहानी हो गई, जो जब ने न मानी तो मैंने भी ठानी, कहा थी मैं देखो कहाँ चली आई। कहते हैं ये दीवानी मस्तानी हो गई। दीवानी हाँ दीवानी दीवानी हो गई। दीवानी 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 हो गई। रूहानी 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 हो गई। रवानी हो गई। मशहूर मेरे इश्क की कहानी हो गई। कहते हैं ये दीवानी मस्तानी हो गई। दीवानी हाँ दीवानी दीवानी हो गई। मशहूर मेरे इश्क की कहानी हो गई, जो जग ने न मानी तो मैंने भी ठानी, कहा थी मैं देखो कहाँ चली आई। कहते हैं ये दीवानी मस्तानी हो गई। दीवानी हाँ दीवानी दीवानी हो गई।
Thank you, Nandita, for your nice performance. এবার আসছে প্রগতি ব্যানার্জি with a beautiful solo dance, PG 4th Sem, Food Science and Nutrition. Please encourage her with a big applause.
किसकी तलवार पर सर रखूं ये बता दो मुझे। इश्क करना अगर खता है तो सजा दो मुझे। ये मोहब्बत के इतिहास लिखने वालों, मैं अगर हरफे गलत हूं तो फिर मिटा दो मुझे।
ईत की लत मोहे ऐसी लागी, हो गई मैं मतवारी, बल बल जाऊं अपने पिया को, हे मैं जाऊं वारी वारी, तेरे नाम से जी लूं, तेरे नाम से मर जाऊं, तेरे नाम से जी लूं, तेरे नाम से मर जाऊं, तेरी जान के सदके में कुछ ऐसा कर जाऊं, तूने क्या कर डाला, मर गई मैं, मिट गई मैं, हो जी हाँ जी, हो गई मैं तेरी दीवानी, दीवानी तेरी दीवानी, दीवानी, तूने क्या कर डाला, मर गई मैं, मिट गई मैं, हो जी हाँ जी, हो गई मैं तेरी दीवानी, दीवानी तेरी दीवानी, चढ़ जाए, इश्क का जादू सर चढ़ कर बोले, इश्क का जादू सर चढ़ कर बोले, खूब लगा लो पहरे रस्ते रब खोले, यही इश्क की मर्जी है, यही रब की मर्जी है, यही इश्क की मर्जी है, यही रब की मर्जी है, तेरे बिन जीना कैसा खुद करती है, तूने क्या कर डाला, मर गई मैं, मिट गई मैं, हो जी हाँ जी, हो गई मैं तेरी दीवानी, दीवानी तेरी दीवानी, दीवानी तेरी दीवानी, दीवानी तेरी दीवानी,
झूमर भी मैं भूल गई, जाने जा तेरे लिए, अरे झुमका भी मैं भूल गई, जाने जा तेरे लिए, अरे मैं तो सब कुछ भूल गई, जाने जा तेरे लिए, तेरे लिए, तेरे लिए, तेरे लिए, तो फिर क्या हुआ, अरे छोड़ छाड़ गए अपने सलीम की गली, ओए होए छोड़ छाड़ गए अपने सलीम की गली, अनारकली डिस्को चली, और छोड़ छाड़ गए अपने सलीम की गली, अनारकली डिस्को चली, अरे देख के तुझको
একটা গান শুনে নেওয়া যাক। মিউজিক, ইমোশন আর বেঙ্গলি সং, এই তিনটে একসাথে মানেই গুজবাম্পস গ্যারেন্টি। সো সিট ব্যাক, রিল্যাক্স অ্যান্ড লেট দ্য মেলোডি টাচ ইন ইউর হার্ট। প্লিজ ওয়েলকাম সত্যজিৎ প্রধান অ্যান্ড সৌমদীপ মাল্লা, ডিপার্টমেন্ট অফ BMLT।
हेलो 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 ভোকালটা একটু কমবে হালকা।
हेलो हेलो हेलो
हेलो हेलो हेलो
মন একে একে দুই একাকার আমি তুই আর না চোখ ভিড়িয়ে একটু হাস নেই মনে কি কিছুই তোর ঠোঁটের ডানা ছুঁই মিলবে সব জীবনের ক্যালকুলাস স্মৃতিরা গেছে পরবাস মালিশে হয়ে ঝুম এ বুকে তো গুবারো মাস ভালোবাসারি ভালোবাসারি মরশুম ডাক নামে ডেকে যাই সেই আগের তোকে চাই সেই যে সেই তাকালে সর্বনাশ ঝড় এলে তুই সাথে থাকলে কি হয় তোর ঠিকানায় পাঠালাম রে হৃদয় প্রেম হলে এক সুরে গান বেজে যায় সে তবু সে ভালোবাসারি মৌসুম, ভালোবাসারি মৌসুম। দিন বদলে যাবে ফের হাত ধরে সময়ে, দেখি মন মাফিক মন পলাশ, দেখি মন মাফিক মন পলাশ। ফুটবে ঠিক মন মাফিক মন পলাশ।
থ্যাংক ইউ সুরজিৎ অ্যান্ড সমজিৎ এত সুন্দর পারফরমেন্স দেওয়ার জন্য। কি মজা আসছে তো? ঠান্ডাটা একটু কম লাগছে? ঘাম বেরোচ্ছে নাকি? এ কি এত ঠান্ডা আবহাওয়া কেন? বুঝেছি, স্টেজটা আর একটু এনার্জেটিক করতে হবে, কি তাই তো? তাহলে চলো আমরা একটু ব্যাকে 90s হিটে যাই, মানে ডুয়েট ডান্স। বলিউড ডুয়েট মানে হিরো হিরোইন ভাইব, মিন্স লাভ, ফ্রেন্ডশিপ, রোম্যান্স, ব্রেকআপ সিন। সো গাইস, সিঙ্গেল থাকলে এক্সট্রা ফিল আর কমিটেড থাকলে ডাবল ফিল। সো ইমোশন ওয়ার্নিং অন। সো প্লিজ ওয়েলকাম প্রীতি অ্যান্ড মোহিত।
तेरी आंखों के मतवाले काजल को मेरा सलाम, जुल्फों के काले काले बादल को मेरा सलाम। हो तेरी आंखों के मतवाले काजल को मेरा सलाम, जुल्फों के काले काले बादल को मेरा सलाम। घायल कर दे मुझे यार तेरी पायल की झनकार, सोनी सोनी तेरी सोनी हर अदा को सलाम। सलाम इश्क 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 सलाम इश्क, सलाम इश्क 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 सलाम इश्क।
दिल का जो हाल है वो तुझे कैसे बयां करे, कह दे तुझे या दिल में रखे बोलो न क्या करे।
दिल जो तुम्हारा है कैसा बेचारा है, मानना बेशर्म बिल्कुल खटारा है।
तू करे दिल बेकरार,
क्यों करूं मैं तुझसे प्यार।
दिल का जो हाल है वो तुझे कैसे बयां करे, कह दे तुझे या दिल में रखे बोलो न क्या करे।
She's so cool, she's so fine. Out of 10, I give her 9.
देखा तुमको जब से बस देखा तुमको यारा, ये देखा तुमको जब से बस देखा तुमको यारा। तुमसे कोई अच्छा है ना तुमसे कोई प्यारा, यूं नजरे ना फेरो तुम मेरे हो मेरे तुम। कह दो ना यू आर माय सोनिया। कह दो ना you are my Sonia।
কি হয়েছে তো? আচ্ছা, নেক্সট অন স্টেজ সাহেলি ঘোষাল from UG 5th Sem Nutrition. She will perform on the soulful song Ore Priya. মঞ্চ ছুঁয়ে যাবে এমন একটা পারফরম্যান্স। So please encourage her.
ओरे पिया ओरे पिया ओरे पिया ओरे पिया उड़ने लगा क्यों मन बाबुला रे आया कहाँ से ये हौसला रे ओरे पिया ओरे पिया मेरे मेरे गा गा मा 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 पा पा मा गा रे सा सा मेरे सा गा गा मा मा गा पा पा मा गा गा मा मेरे सा गा मा पा ला मा दा रे दा रे दा वाना वाना ताना वाना घूमते हवा है घूमती हवा बूंदे भी तो आए नहीं बाज यहाँ साजिश में शामिल सारा जहाँ है हर जर्रे जर्रे की ये इल्तिजा है ओरे पिया
थैंक यू। टू डांसर वन स्टेज मतलब डबल धमाका। कोऑर्डिनेशन एनर्जी एंड केमिस्ट्री इक्वल टू শ্রেয়া দে এন্ড রূপসা দাস বিএমএম 5th সেম সব একসাথে দেখার জন্য রেডি তো? লেটস চেয়ার দেম আপ। এরপর অদিতি চক্রবর্তী রেডি থাকবে, চলে আসো এখানে।
नैन मोरे तरस गई रे आजा बलम परदेसी नैन मोरे तरस गई रे आजा बालम परदेसी পানসা জলে সাই ভাষায় সাগরের বানে আমি জীবনের ভেলা ভাসাইলাম কেউ না তা জানে রে
অকুল দরিয়ায় বুঝি কুল নাই রে আমায় ডুবাইলি রে আমায় ভাসাইলি রে অকুল দরিয়ায় বুঝি কুল নাই রে
साजन मोरे घर आवे
साजन मोरे घर आवे आत्मन सुख पावे
आत्मन सुख पावे
साजन मोरे घर आवे साजन मोरे घर आवे
ভেঙে মোর ঘরের চাবি নিয়ে যাবি কে আমারে
ভেঙে মোর ঘরের চাবি নিয়ে যাবি কে আমারে
ও বন্ধু আমার না পেয়ে তোমার দেখা একা একা দিন যে আমার কাটে না রে
ভেঙে মোর ঘরের চাবি নিয়ে যাবি কে আমারে ভেঙে মোর ঘরের চাবি নিয়ে যাবি কে আমারে
জয় রাধার নামে পাগল হয়ে থাকবো মহগোনা জয় রাধার নামে পাগল হয়ে থাকবো মহগোনা
না 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 ছেড়ে দেব না
তোমায় হৃদ মাঝারে রাখিব ছেড়ে দেব না তোমায় হৃদয় মাঝে রাখিব ছেড়ে দেব না
অসংখ্য ধন্যবাদ শ্রেয়া ও রূপসাকে। এবার কিন্তু একটু কথা বলা বন্ধ করতে হবে, কারণ এবার হার্ট ফিল করবে। এখন স্টেজে আসছে আমাদের সবার কাছে সাইয়ারা, অদিতি চক্রবর্তী, ইউজি 5th সেম নিউট্রিশন।
सांसों के साज में हूँ, आंखें झुका के सुन ले मुझे तो, मैं तेरे दिल की आवाज में हूँ। तू हौसला है, तू है इरादा, आधी मैं तुझ में, मुझ में तो आधा। टूटू न मैं भी, टूटे न तू भी, मैं तेरा सपना, तू मेरा वादा। हाय मैं मर ही जाऊं जो तुझको ना पाऊं, बातों में तेरी मैं रातें बिताऊं। होठों पे लम्हा लम्हा है नाम तेरा है, तुझको ही गाऊं मैं, तुझको पुकारूं।
আবারও একটা ফোক ডান্স দেখে নেব। ফোক ডান্স মানেই আমাদের শিকড়, কালচার আর ট্রেডিশনের গল্প। নেক্সট অন স্টেজ বর্ণালী বেরা ফ্রম ইউজি থার্ড সেম ডিপার্টমেন্ট অফ নিউট্রিশন।
ও রঙ্গবতী রে রঙ্গবতী, আরে রঙ্গবতী রঙ্গবতী কনকলতা, হসি পদে কহন কথা।
হায় গো লাজে 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 লাজে, হে লাজে 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 লাজে, লাজে লাজে লাই যাଉଛି মাথা মোর, নাই করো অথা।
আরে মিত্র ভানু গন্তিয়ার কথায় প্রভু দত্ত প্রধান করিল সুর। এত সুন্দর গীত আমাদের, এই গীত একটু তোমাদের ভাষায় শোনাবো নাকি গো? রঙ্গবতী রে রঙ্গবতী, ও রঙ্গবতী রে রঙ্গবতী, রঙ্গবতী রঙ্গবতী রঙ্গিলা অন্তরে হাবুডুবু তোমারি কথা। রঙ্গবতী রঙ্গবতী রঙ্গিলা অন্তরে হাবুডুবু তোমারি কথা।
হায় গো লাজে লাজে লাজে, হায় গো লাজে লাজে লাজে, হায় লাজে লাজে মরে দোহাই রসিক নাগর মনে নাই কোনো ব্যথা। লাজে লাজে মরে দোহাই রসিক নাগর মনে নাই কোনো ব্যথা।
স্বপ্ন দিলে স্বপ্ন,
স্বপ্ন আবার দিলে স্বপ্ন,
রূপে তোমার মরি বাছি বক্ষ জুড়ে হাহাকার। রঙ্গবতী রঙ্গবতী, ও রঙ্গবতী রে রঙ্গবতী, ও রঙ্গবতী রে রঙ্গবতী। প্রিয়া প্রিয়ার নলক হাতে কাকন, মন বিহঙ্গন আমার জীবন ধুলায় উড়ি যায় রে। প্রিয়া প্রিয়ার নলক হাতে কাকন, মন বিহঙ্গন আমার জীবন ধুলায় উড়ি 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 যায় রে।
মুখে কোনো কথা নাই যে গো, মুখে কোনো কথা নাই যে গো, মুখে কোনো কথা নাই, শুধু আকারে সাকারে আখির বাহারে সরষার খেতে দুইজন গো। মন আকারে সাকারে আখির বাহারে সরষার খেতে ভাসে গো।
ডুবে 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 আমার অন্তর পুড়িল, ডুবে 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 আমার অন্তর পুড়িল।
কুহ কুহ ডাকে আবার বুকের মাঝে হয়তো
রঙ্গবতী রঙ্গবতী, ও রঙ্গবতী রে রঙ্গবতী, ও রঙ্গবতী রে রঙ্গবতী। হায় রঙ্গবতী রঙ্গবতী রঙ্গিলা অন্তরে হাবুডুবু তোমারি কথা। রঙ্গবতী রঙ্গবতী রঙ্গিলা অন্তরে হাবুডুবু তোমারি কথা।
হায় গো লাজে লাজে লাজে, হায় গো লাজে লাজে লাজে, হায় লাজে লাজে মরে দোহাই রসিক নগর মনে নাই কোনো ব্যথা।
হ্যালো ছেলে মেয়ে যারা একসঙ্গে ডান্স করছে ওরা সেপারেট হয়ে যাও,
হসি পদে
ডান্স করো সেপারেট।
রঙ্গবতী রঙ্গবতী কনকলতা, হসি পদে কহন কথা। রঙ্গবতী রঙ্গবতী কনকলতা, হসি পদে কহন কথা।
Hello. Thank you. Next performer Rajashree Majhi from UG third SEM Department of Nutrition. Thank you, Rajashri. Next performer, Deepti Pramanik from BMM 5th SEM with a beautiful solo dance. Please welcome her with a warm applause.
धुमकिट तदानि धुमकिट नटना गरनट धिम नट पर्वत उभा उत्तुंग नवा घुमतो मृदुंग. धुमकिट तदानि धुमकिट नटनागर नट धिम नट पर्वत उभा उत्तुंग नवा घुमतो मृदुंग पखवाज देता आवाज जनन झंकार लेऊनी स्त्री रूप बुलवी नटरंग नटरंग नटरंग
रसिक होऊ दे दंग चढू दे रंग असा खेळाला साता जन्माची देवा पुण्याई लागू दे आज पणाला आज जोड आज आम्हाला रंग तुझा दे गलकारी नवा स्वताल गावले
থ্যাংক ইউ দীপ্তি। অনুষ্ঠান প্রায় শেষের দিকে। আমার সঙ্গী অনেকক্ষণ ধরে আমার কাছে দাঁড়িয়ে এই নাচের তালে তালে নাচছিল। এখন ওরই একটি পারফরমেন্স দেখে নেব। সো লেটস ওয়েলকাম মাম্পি মান্না ফ্রম 4th সেমিস্টার ফুড সায়েন্স অ্যান্ড নিউট্রিশন।
से नैनों को मिला, नैन से नैनों को मिला, नैन से नैनों को मिला, नैन से नैनों को मिला, काहे को सताए हैं अभी जा, देखना यूं बेरुखी से आज तो नजर मिला ले, है कसम तुझे दीवानी प्यार का कोई सिला ले, जब से तेरी आंखें झुकी है तब से मेरी सांसें रुकी है, चोरी चोरी ख्वाबों में अभी जा, नैन से नैनों को मिला, नैन से नैनों को मिला, काहे को सताए हैं अभी जा।
चलिया 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 रूह चुरा लूं मैं हूँ ऐसी छलिया, चलिया चलिया ओ चलिया, हाथ ना आऊं मैं हूँ ऐसी छलिया। चलिया 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 रूह चुरा लूं मैं हूँ ऐसी छलिया, चलिया चलिया ओ चलिया, हाथ ना आऊं मैं हूँ ऐसी छलिया। ओस लहर हूँ धूप सहर हूँ पाक महर हूँ, बच के जाएगा कहाँ, ना लड़ा तू अपना लड़ा, don't look at me like that, माही ना लड़ा तू अपना लड़ा, don't look at me like that। कच्ची कली साडा मासूम चेहरा, नीयत नूरानी साडा दिल है सुनहरा, कच्ची कली साडा मासूम चेहरा, नीयत नूरानी साडा दिल है सुनहरा। पर्दे दे पीछे की है किसनू पता, पर्दे दे पीछे की है किसनू पता, खफीमी मैं चस्का चखामा, तैनू शरारत सिखामा, जदों नैना लड़ावा, तैनू शरारत सिखावा, जदों नैना लड़ावा। नैना च मेरे एक नीला समंदर, नैना च मेरे एक नीला समंदर, समंदर में डुबकी लवावा, तैनू शरारत सिखावा, जदों नैना लड़ावा, तैनू शरारत सिखावा, जदों नैना लड़ावा।
Thank you, Mampi. And now, the moment everyone was waiting for. This is not just a performance. This is a celebration of unity, friendship, and energy. Presenting the group dance, Bengali Mix. I mean, our favorite Dev. Performed by UG student of Nutrition, Zoology, and Chemistry. Different departments, one stage, one vibe. So please put your hands together, cheer loudly, and enjoy the grand final. কি সবাই রেডি তো দেব ফেভারের জন্য? আর একটু জোর, শেষের দিকে এবার। ওকে চলো রেডি, এবার দেখে নিই একটু দেব ফিভার। চলো দেখাও তোমাদের মধ্যে দেবের ফিভারটা ঠিক কেমন।
বলে বলুক রে তাল বেতালে হেলে দুলে কোমরটা দুলুক রে রাস্তা জালে উঠোন বাঁকা যে বলে বলুক রে তাল বেতালে হেলে দুলে কোমরটা দুলুক রে লে চান্স রে লে চান্স রে লে চান্স 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 লে পাগলু ডান্স le pagaloo dance le pagaloo dance le pagaloo dance
हाय चुमकी रखी खवाली धुमकी दे आज बवाली चुमकी रखी खवाली धुमकी दे आज बवाली एक और नशा धराली गायचे ए दिल कवाली प्रेमर आचे बुकरे माजे लागलो जे छक्का लगा 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 प्यार का झटका अरे लगा 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 प्यार का झटका अरे लगा 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 प्यार का झटका अरे लगा 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 प्यार का झटका
Kolkata, get down tonight. Bojo Gorango to all the ladies.
get down tonight the beats are right Bojo Gorango let's get it on now Bojo Gorango let's get it on now down tonight the beats are right Bojo Gorango Bojo Gorango Bojo Gorango Cristo Ole Lila Olila 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 Amra Ole Bila Olila 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 Caro Cristo Ole Lila Amra Ole Bila What do you get this torada What do you get this torada What do you get this torada Disco technique conectan Bojo Gorango Logo Ranger Navre Bojo Gorango Logo Ranger Navre Bojo Gorango Logo Ranger Navre Bojo Gorango Logo Ranger Navre
মনের যে নাই কোনো কাম ও বন্ধু মনের যে নাই কোনো কাম এই মনের যে নাই কোনো কাম ও বন্ধু মনের যে নাই কোনো কাম তোর পিড়িতে দিবানিশি থাকে আমার মন উদাসী প্রেমের জ্বালা বড়ই জ্বালা রে এই বুকের মাঝে আগুন জ্বলে নেবে না রে জ্বলছে গালে কবে দিমু গলায় মালা রে আরে কবে 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 আইবে পালা রে আরে দিহি মুহু গলায় মালা রে মালা রে মালা রে কবে আইবে আমার পালা রে কবে দিহি মুহু গলায় মালা রে মালা রে মালা রে
ভারত আমাদের দেশ, এখানে প্রতিটি অঞ্চল।
থ্যাংক ইউ এভরিওয়ান। কি সবার দেব ফিভার কাটলো তো? As we come to the end of this cultural evening, we sincerely thank you so much for the performance organizers and respected teachers. সেমিনার আমাদের শেখায় আর কালচারাল প্রোগ্রাম আমাদের মনে করিয়ে দেয় জয়, ক্রিয়েটিভিটি and togetherness are equally important in our academic life. So thank you everyone. Yes, have a wonderful time. Stay happy, stay healthy.